we're sorted out with. And thank you so much for coming today. I'm going to ask Dr. Stein Brown, who's the Dean of the Dalai Lama School of Public Health and who is very much responsible for the existence of the Center for Vaccine Preventable Diseases at U of T, who is a driving force who made it happen. Thank you, Stein, for that. I'd like you to make some opening remarks, please.
So thanks, Natasha. Look, I'll just make a couple, 3 remarks, and I won't pretend to delve into the science or somehow encapsulate what will be discussed today. But if you're like me and you're interested in things like public health, these are days that kind of give you a few sleepless nights. And so probably like a lot of you, I read a lot too at night. And I've been reading recently the biography of Rockefeller. And I don't know if anyone knows about Rockefeller. It's kind of an interesting biography. The first 2/3 is interesting business practices, that's probably the diplomatic term, and huge amounts of money accumulated. And it's actually kind of a boring book for the first little bit. But then there's this kind of moment when things shift. And I don't know what his moment is, where he changes what he's doing, but everything kind of changes wholesale. He becomes a happy person. He'd actually lost all his hair. He starts to grow his hair back, interestingly enough. He starts to spend time with his grandchildren. And he starts to put incredible support behind medical research. And not just through the Rockefeller Institute or the Rockefeller University now, but it shifts from being a story about one person to being a story about a whole team of people that really had relatively little to do about him, with him. And you can see things like hookworm disappear out of entire states, things that had caused almost a crushing, physical and economic burden on an entire state here, an entire country there, just disappear. But it's not because someone discovered a cure. It's not because someone figured out how to manufacture that cure, or even the kind of, sometimes we think, boring idea of how to distribute that and the logistics of it. It's not because someone sat around and wrote a series of papers or articles or was in the press or constantly out pushing why they had to deal with things like hookworm. It's because all of it comes together. And this is something I think sometimes we forget when we look at the big changes that really improve health. It's easy to tell these as stories of an individual hero, and I think we all kind of like that and can see that as a great narrative. But it's always in these sorts of things a team sport. And so I'm very proud to be the dean of the School of Public Health. I get this moment of, I think, probably call it sort of false pride or false ownership when Natasha says something nice and says he's been the driving force. For those of you who are considering a career in academic administration, that means you get to talk at the beginning of interesting days like this and immediately leave. And you get to put a little bit of resource behind really good ideas. But it is really very much a team sport. And the whole founding of the school, its start really dealing with antitoxin, its work later on things like polio vaccines and making sure they were within reach of all. I think that's actually kind of the subtitle of the book that Chris Ruddy wrote about our school. It's health within reach of all, right? And so that's really a team sport. And so when I look at the agenda today, the first half of the day is about all the basic science and how we get better and better vaccines. Second half of the day is how we make sure that those vaccines get used and taken up. And both, in our current environment, are really critical questions. And so I'm really excited to see a room that is filled to capacity. You always worry that you're going to have a lot of leftover catering. That's not going to be the problem today. I'm really excited to see such a diverse group. I'm really proud that the Dalai Lama gets to support this, but I'm really glad that there's such a broad team of people here. And I'm really grateful to Natasha, to Jeff, and to others who've had a real leadership role in moving this along. But I'm really pleased to see everyone here today. And thanks, and have a great day. Natasha?
Thanks, Stein. And I was serious about him being the driving force. It wouldn't have happened without him, I have to say. But I totally echo the remarks about the team sport. Today, you may have heard all the news about the centre and wondered what it was about. There have been so many people involved in the initiation of the idea back in the days when Shelley and I said we need a centre in Ontario. That was probably a decade ago. All the way through to more recent times, and many of you are in the room and you know who you are. And thanks to Jeff, who's agreed to be, you're sitting there, aren't you? The Associate Director of the Centre, so that's good news. It really all starts with the faculty and the students, and a lot of the faculty and the students are here as well, and that's the goal of the centre, to reach and bring together people and move things forward. So I'm hoping today will be a big part of that. The faculty are networked to a number of people outside in organisations, and so the team is really quite a complex team, reaching far and wide outside the university. My home institution, Public Health Ontario, has been incredibly important in driving this day forward and in driving this whole initiative forward. So we have to recognise Peter Donnelly, who's been thoroughly behind it as well, who's our CEO and president. I didn't actually say who I am. I'm Natasha Crowcroft, and I'm, in case any of you don't know, I'm the Chief Science Officer at Public Health Ontario and also a Profit UAT and an adjunct scientist at ICES. So in the spirit of teamwork, the morning is going to be chaired by one of my dear colleagues from Public Health Ontario, Dr. Bryna Wachowski, who's the Medical Director of Communicable Diseases Emergency Preparedness and Response Department at Public Health Ontario. And she's also cross-appointed to the Department of Epidemiology and Biostatistics at Western University, and a past chair of the National Advisory Committee on Immunisation, which is an incredibly important role in Canada. So we're very lucky to have her to chair the morning. So Bryna, I'd like to hand over to you, if you don't mind. Thank you.
Thank you very much, Natasha. Thank you for the opportunity to chair this morning. It's a real privilege to be here. It's so great that the room is so packed with so many people who are interested in vaccine. That's fabulous. So we're going to start our first presentation by heading across the ocean via video teleconference, and we're glad that hopefully this will work. So it's my pleasure to introduce Dr. Gunstein Norheim. Dr. Norheim is the Director for Vaccine Sciences at the Coalition for Epidemic Preparedness Innovations, and he was central to the development of this organization. He has a PhD in molecular epidemiology and meningococcal vaccine development from the University of Oslo, and in 2007 as part of the vaccine development environment at the Norwegian Institute of Public Health. He worked as a consultant for the WHO in Burkina Faso and as a postdoctoral researcher at the University of Oxford. Until 2018, he was employed as a senior scientist at the Norwegian Institute of Public Health, working on infectious diseases and vaccine development, including meningococcal and tuberculosis, particularly focusing on developing countries. And in 2014, he was part of the international team that developed the phase 3 clinical trial for the Ebola vaccine in Guinea. So we're very honored to have Dr. Norheim present via teleconference, and I'll turn it over to you. Thank you.
Thank you. Hi, I'm Stein. Thank you for inviting me to this important conference. My background is coming from vaccine development, but this new job is a task that takes a lot more than a few individuals. It takes a community of scientists and experts in all countries affected by outbreaks of emerging infectious diseases. So we are very much trying to build an international coalition. Our goal is to, let me just share these slides here. First, I'd like to state that I have no conflicts to report. Going on to the roots of CEPI, we really need a global coalition to address this challenge of the emerging infectious disease outbreaks. And we set out in 2016 together with and based on a mandate through the WHO blueprint for R&D, where a series of recommendations were made how the system should be set up or fixed to make sure that we are better prepared next time, so that there is no disfragmented response, or that there is actually a prospective planning towards working on outbreaks and vaccine development. Okay, just a second. Can you see them?
Yeah, we can see them now. Thank you.
Okay, good. Very good. So we have a coalition now built with over 10 sovereign states and private foundations, and we are working towards building more donors. Canada is one of them. I'll come back to the role of Canada at the end. On the right hand here, you can see the group that launched CEPI. It's quite strong mandated, a link to the WHO blueprint and role of how we all together should work to prevent new outbreaks. So the vision is a world which epidemics are no longer a threat to humanity. This is a big wish. We do one step at a time, and we work by accelerating the development of vaccines against emerging infectious diseases and enables equitable access to those vaccines for affected population during outbreaks. And as we speak, you know the outbreak in DRC, which is affected by multiple factors, some of which are medical, some are political. CEPI is working to address the medical needs here, and we are specifically focusing on vaccines as part of that. So we have 3 strategic objectives during the 5-year period. We work on preparedness by advancing current vaccines towards a next level, towards phase 2 and further on. We work on preparing the response so that it can be accelerated, and we work on systems and the end-to-end picture to make these vaccines accessible also over time. And in order to make this a sustainable partnership, we're not working alone. We work either as a funder directly into projects, or we work as a facilitator where we collaborate with others where there are complementary strengths and try to encourage funding where there are gaps that we see are directly linked or relevant for vaccine development. So CEPI, what have we set out to achieve in our first 5 years? First, we would like to develop 2 to 3 vaccines for 2 to 3 priority pathogens until end of phase 2. And we will initiate a response for vaccine development to respond to new outbreaks. And this means investing in vaccine technology platforms that are versatile and can be used with a lot shorter time for development than traditionally is now. And third, we'd like to create a coalition where there is a community responsible for this development over time. Some of you might be familiar with this list. This is a blueprint list for target pathogens that WHO now recommends there should be intensified R&D for. And out of this, CEPI is working on a series of them. There's also a watch list below here on the slide, and we are taking on some of them as well. From that list, we worked on finding out which of them had a feasible vaccine candidate being developed and enabled to reach phase 2 by 5 years, which of the diseases and for which vaccines, for which diseases would a vaccine have a significant health impact. And these are the diseases that we start out with: MERS, Lassa, and Nipah. And in the last year, we've started with chikungunya and Rift Valley fever. We are also working towards disease X, which is the disease platform concept. I'll come back to that in a minute. Some of these diseases are sporadic, some of them are endemic, and some are quite high case fatality rates, some are very low and have a disease burden that is different from another. So for Lassa fever, it's mainly affecting 5 to 8 countries in West Africa with an annual disease burden peaking in January to March, and the outbreak in Nigeria last year and this year have been very much noted and higher than it has been before. It could also be explained by intensified surveillance, but more than 550 human cases were confirmed this year. For MERS, this is another sporadic disease affecting mainly camel herd communities, and the cases are detected in countries, particularly Saudi Arabia. However, the surveillance system for MERS, given that this is a quite sporadic disease, is also fragmented across. As well, there are gaps in diagnostics and how these are used. For Nipah, outbreaks have been noted, especially in the last year in Kerala in India, very high case fatality rate, and the potential for spread is what is worrying. We do not know how wide it will spread, but there is the host, which is a very wide range of spread, but the noted outbreaks are in 3 to 4 countries, and we know that to work in this setting, we need to be prepared, and therefore a Nipah vaccine is now being prioritized.
Dr.
Rift Valley
Norham,
fever.
can I interrupt you just for 1 minute? I've been informed that it's hard to see in the back because I think you're in speaker mode, and if you could change the view to presentation mode so that we can see the full slides on the screen, is that possible?
Okay.
Thank you.
Sure. Good. So we are working on Rift Valley fever. Human cases are occurring in several countries, and about 4,000 cases noted the last 20 years. Chikungunya is a lot more widespread. Case fatality rate is low, but the disease burden in terms of arthritis is significant and causing an effect on mobility, ability to work, and income of families. So for our ways of including vaccines for this, we have launched open calls. We have received applications worldwide from this, biotechs, big pharma, and PDPs, universities. And from this, we work towards the goal of having a phase 2 trial done, as well as a stockpile of 100,000 doses ready for outbreak at the end of 5 years. So each of the CFP1 or the MERS, Lassa, Nipah developers have this goal in their contract. Secondly, for disease X, we know that there is a wide variety of pathogens that could cause outbreaks. However, the epidemic hotspots are limited to some geographic areas, mainly high density populations and close interaction with human and animals. So this is the area that we should focus on and where we think that future outbreaks will occur. And the important thing is how fast can we respond with developing a vaccine in time to be able to mitigate an outbreak. So for that, we are aspiring to reduce time, 16 weeks from identification of a pathogen until a GP product is ready, 6 weeks from first dose until a clinical benefit, 8 weeks for manufacturing 100,000 doses. So these are aspirational goals, and some technologies manage it, some don't. Secondly, we would want these to be proven that they are versatile, that they could provide protection across 3 different pathogens, and out of these 2 will be tested in a phase 1. So these data will be helpful in validating or proof testing whether these platforms are suitable as part of a toolbox of different technologies towards new outbreaks of new pathogens. Here you can see a list of the typical technologies we've been working on. Some of them have, they each have their strength and weakness, and they will each be useful in their setting. So this is the list of our partnerships to date. We have funding of around $720 million over this 5-year period. We're working towards recruiting $1 billion, and we are investing them in projects like this. Here you can see our current portfolio. The blue boxes are those that are based on viral vectors. You can see the VSV, the role of the Public Health Agency Canada is important in this regard, or in the origin of proving that, proof of concept. Others are measles and adenovirus. We invest in subunit proteins and in nucleic acid-based vaccines. And we have 21 vaccine candidates with 11 different partnerships as we speak. And we are now working on signing with signing contracts for up to $48 million in total for chikungunya and Rift Valley fever mid-year. We work with WHO, the countries. We work on finding what data are missing on epi and diagnostics. We work to engage regulatory agencies to support this development, and we work with a long-term view of how can this contribute to a licensure in affected countries. We therefore also have a wider scope only than the vaccine development project. We also work on epi project, and we are now launching a 5-country Lassa epi study in West Africa. We work with Find on Diagnostics to support their work so that addresses Lassa. We work with the developers and regulatory agencies and laboratories on assay standards and animal models, and we will work on the trial capacity in affected countries with those that are best at this and suiting the purpose of future vaccine trials. We also work with Brighton collaboration on safety monitoring. So finally, the role of Canada and CEPI is a very critical partnership. As Canada is a coalition member, did its first investment in 2017, and it did a second investment in 2018, announced by Trudeau. Secondly, Canada has a tremendous amount of expertise. You're recognized in disease surveillance and control, both in human and animal health. You have large recognized institutions that are research intense, and we have used your experts in review of these candidates. So we approached the issue of recruiting candidates in a way that we want the best experts to take part and assess these candidates in a peer review manner, so that vaccine candidate developers can get the best information and constructively improve their development plans. We also have a multitude of vaccine research centers relevant for future clinical development. And we want Canadian actors as part of awardee consortia, and there will be more and forthcoming announcement for and opportunities for this. So we look very much forward with a continued partnership with Canada and the expertise in vaccine development. Thank you.
Thank you very much, Dr. Nordstrom, for describing the very important work that CEPI is doing. So we do have about 10 minutes for questions, and I will try to translate the questions to make sure that Dr. Nordstrom can hear them. Are there questions in the room about the work that Dr. Nordstrom has described, or new vaccine development? Yeah.
I'm just curious because it's very expensive to develop a new vaccine. So what is the estimate cost of getting one of these new ones before it's due?
So the question from Dr. Watts was, what is the cost of getting one of these new vaccines to implementation, given that developing new technologies can be expensive?
So we are working so that cost is, of course, a central question. It depends on who is engaged in that and the cost of goods to the full end. And we don't have one set figure for that from this work. We see that the costs are differing a lot across actors, and depending on what the endpoint is. If you work we have only funded project until end of phase 2, and that's not the end of it. We also have, maybe I can show a slide here in the end, which shows which part are we actually not having funding for. And that leads to your question on what is the cost of leading it to licensure. You see the box on the left, this is what we have funding for. It's non-clinical proof of concept, phase 1 and phase 2, and manufacturing for the stockpile. But the box on the right is everything that you need to get this through to licensure, and that is not funded.
Okay.
And I like to refer you to a paper by Dimitris Gouglas in Lancet Global Health that looks into the costs of development. The reason he's a CEPI colleague that has tried to estimate this. Thank you.
Thank you. So, other questions in the room? So I had a question, just in terms of the technologies, which would you think is the most prominent, most likely to be successful in the near term?
Yeah, so we are also wondering of that, and therefore we are applying a portfolio approach with, for example, for Lassa, we have 5 candidates. We have a DNA vaccine, we have several viral vector vaccines, and we have an mRNA candidate. And the reason that we go this way is that the correlate of protection for Lassa is not established. It could be that cellular immunity is most important. It could be that neutralizing antibodies is the correlate or the mechanism for protection. So as long as that is not established, we need to pursue several candidates, both for that reason, but also for other reasons for product success. There's no single solution to all of these diseases. They all have their own likely correlated protection. Very often neutralizing antibodies is key, but might also not be enough.
Thank you. Other questions in the room?
Would 100 million be a reasonable figure as an estimate?
So the question was, will 100 million be a reasonable figure as an estimate?
It's hard. It's hard to say. It's hard to say. It will help, for sure.
Natasha? Oh,
I think Tanya had a follow-up.
okay, Tanya, go ahead.
I just had another question about with the development of these new vaccine candidates to phase 2. I'm wondering if you also have funding to do immunophenotyping and correlates of protection in the clinical trials.
So the question was, with the progression of some of these vaccines to phase 2, whether there is funding to do immunophenotyping?
Yeah, so each of these are asked to do as much as they can of a correlative protection study, meaning bridging the NHP or like animal protection study immune response with the response seen in humans. So we will develop and fund a lot of experiments that will prove useful over time, and we are working on a concept of correlative protection studies across diseases that will come later on. So there have to be more work on this, especially for diseases where there is not enough cases to justify an efficacy trial. For example, for Nipah, it's very hard to be ready in time at the place when an outbreak occurs with a setting where phase 3 trial can occur. So most likely licensure have to be based on a correlative protection, establishing immune correlates in animals first, and then bridging that to immune responses in clinical trials. Over.
Thank you. And we have time for one final question. I'm noting there is a microphone for people who want to get up and ask the questions where Natasha's standing. So.
Okay, thanks. I actually had 2 questions, but it might be a bit tricky. The first one might be quick. I think I heard you say you're working on a measles vaccine, or am I just hallucinating measles all the time?
Could you hear that question?
No, I noticed it was about measles.
Yeah, the
Sorry,
question,
could you repeat
are you
that?
working on a measles vaccine, or is Natasha hallucinating?
Yeah. No, the key with the measles vaccine concept is the pre-existing immunity and how that can be overcome, and whether it complements and can provide protection for measles. So there's a few papers on that from this developer, it's Temis. So I'd like to refer you to that for further info.
Okay, so we'll have to look that up. Thank you, that was a surprise. And the second question I had, if it's okay, it might be hard for you to answer, but it was, you mentioned the outbreak of Ebola in the Democratic Republic of Congo. And the big challenge there is not about the vaccine, we have a vaccine, which is a miracle now, but the public confidence. And that's with a vaccine that has actually had the trial, as you know very well, better than anyone in West Africa. So not going in with a completely new vaccine. So I just wondered how CEPI is thinking about this new technology and these new vaccines that are going to be developed. How are you thinking about preparing communities, given the experience in countries like DRC, of the kind of community response even when you have a very life-threatening disease, communities may not accept a vaccine and may indeed turn on the very people who are trying to help them. Thanks.
Yeah. No, I think the key is involvement early on in development, all the way from prioritization of pathogens down to when you do epi studies, why you do those, trying to argue for why science-based decisions are likely to be successful, and also how communities are engaged in epi studies, but also in trials, of course, and down to the implementation in an outbreak. So it's clear from the DRC outbreak that the community resistance is based on distrust that probably has more background to it with previous history of the area than only the vaccine trials. So it's extremely important to be kind of location or knowledgeable about the local situation where the trials will be, what issues they are sensitive to. But involvement early on is absolute.
Great. Thank you very much. Thank you. So if you can join me in thanking Dr. Norm for a very interesting presentation.
Thank
We
you.
really appreciate you joining us and look forward to hearing about the future work of CEPI. So thank you. Okay, so our next two presentations are a panel discussion, and they will focus on new technologies and vaccines. And we're fortunate to have two speakers, Dr. Epshida Islam and Dr. Keith Parody. So what I'll do is I'll introduce Dr. Islam first, and then she can do her presentation, and then I'll ask her to sit over here, and then we'll go to Dr. Parody. So I'm going to just apologize up front. I'm going to shorten the bios for these people who have many accomplishments, but just in the interest of time, I'll just focus on the key points to give them as much time as possible for their presentations. So, Dr. Epshida Islam is a MyTech a postdoctoral fellow in the Department of Biochemistry at the University of Toronto. She manages a multi-institutional collaborative research program aimed at developing novel vaccines against several important human and veterinary bacterial pathogens. And she's going to describe her very interesting and important research. So I invite Dr. Islam to come to the podium.
Good morning, everyone, and I would like to thank the organizers for giving me the opportunity to speak today.
Not as short as me. No one's as short as me.
Thank you. Originally, my supervisor, Dr. Scott Rayon, was supposed to give this talk. However, he had an emergency, so he apologizes that he wasn't able to make it today. So hopefully I can walk you through some of the vaccine development technologies that we're developing in the lab. I have no affiliations to disclose. So at the heart of our vaccine development platform lies the importance of iron. Iron is an essential micronutrient that is required for all forms of life, from humans all the way down to microorganisms, and this includes disease-causing pathogens as well. As a result of this, free iron bioavailability in our bodies is actually very tightly regulated by high-affinity iron-binding proteins in blood as well as on mucosal surfaces. And the major iron-binding protein is a protein that's called transferrin. So in a way, by limiting the bioavailability of free iron, these iron-binding proteins actually have a protective effect against infections, and this is known as nutritional immunity. However, there are pathogens of humans that have evolved ways to extract iron from this iron-binding protein called transferrin in order to promote its own growth. So these pathogens include pathogens belonging to the Neisseria family, so Neisseria meningitidis, that causes meningitis and septicemia, Neisseria gonorrhea, which causes the sexually transmitted infection, gonorrhea. Also, there's Haemophilus influenzae and Moraxella catarrhalis, which cause otitis media, lower respiratory tract infections, and can also cause meningitis. So the way that these pathogens are able to acquire iron from human transferrin is through the bacterial transferrin receptor. So it's a two-component receptor system that consists of an outer membrane lipoprotein called TBPB, which is shown in green in this ribbon diagram, and a integral outer membrane protein called TBPA, which is shown in purple in this diagram. So the way that the transferrin receptor works is that TBPB extends out of the surface of the bacteria and grabs iron-loaded transferrin and brings it to TBPA, which is then able to extract the iron from transferrin and transport it to the inside of the bacterial cell for its growth. So we asked the question whether we could utilize this transferrin receptor as a vaccine target. And we chose to focus on pathogenic Neisseria because of the importance of Neisseria gonorrhea, and developing a vaccine for it has become essential because of the rise in antibiotic resistance in this pathogen. And we chose to focus on the outer membrane lipoprotein, TBPB, as the vaccine antigen, because it's an extremely stable protein and it's easy to recombinantly produce it in E. coli and purify it. So from a commercial point of view, it's a better target to work with. So we evaluated the effect of TBPB on Neisseria infections using mouse models. So the first study that we did was using a sepsis model of Neisseria meningitis, where we infected mice with a lethal dose of bacteria. And what we found was that the adjuvant controls were all reaching endpoint very early, whereas the TBPB immunized animals were all protected 100%. And as a positive control, we used factor H binding protein, which is an antigen that's found in the two licensed Group B meningococcal vaccines. So we also evaluated TBPB in a nasal colonization model of Neisseria meningitis. So in this model, we utilize a human CCAM1 mouse, which is the mucosal receptor for Neisseria meningitis in the nasal cavity. And we challenge these mice intranasally with Neisseria meningitis and look at bacterial recovery 3 days post-infection. And what we found in this model was that compared to the adjuvant control, TBPB immunized animals were able to reduce the colonization rates significantly. And this was very interesting because the factor H binding protein antigen did not have this effect. Next, we also evaluated TBPB in a mouse model of Neisseria gonorrhea infection. So in this model, female mice that have been treated with hormones and antibiotics to enable gonococcal colonization are infected in the lower genital tract, and we monitored the duration of infection by looking at bacterial recovery from daily vaginal lavages. And in this model, what we found was that compared to the control, the TBPB immunized animals were also able to clear infection much faster. So overall, it looks like TBPB was able to confer protection against not only just invasive infections by pathogenic Neisseria, but also able to confer protection on the mucosal surface. So these studies were very, very promising. However, one of the caveats of these previous studies was that they didn't take into account the specificity of the transferrin binding protein B antigen and that of human transferrin. So what we were seeing in the previous studies was this sort of a scenario. So TBPB is highly, highly specific for human transferrin and does not bind mouse transferrin. So in the previous studies, when we immunized with TBPB antigen, it did not recognize the native host transferrin, which was mouse transferrin in that case, and all of its epitopes were exposed. However, if we were to take this antigen into its native host, that is humans, we would expect that this scenario might occur, where the antigen itself would bind to the human transferrin that's present in blood, because human transferrin is present in great abundance, and that would result in some of its epitopes being hidden from the immune system. So in a way to overcome this potential issue, we decided that we wanted to generate non-binding mutants of the TBPB antigen that will not be able to bind the native host transferrin. So the way that we went about this is we solved high-resolution crystal structures of TBPB and identified residues that were important in mediating the interaction with human transferrin. And then we were able to mutate a single amino acid that completely abolished the interaction with the human transferrin. So if we use this antigen, the non-binding TBPB antigen, in the presence of human transferrin, we expected that, we hypothesized that this would be a superior antigen in mediating protection compared to the wild type. So because we can't really do these studies in humans, we again made use of animal models. So we utilized human transgenic, human transferrin transgenic mouse model So in this model, human transferrin is expressed at physiological levels to that in humans. And we utilized a similar gonococcal colonization study, as I've shown before. And what we found was that indeed the non-binding TBPB mutant, upon immunization, was able to clear infection much faster than the group that was immunized with the native TBPB antigen. So we wondered why we are seeing this increased protection. So we wanted to evaluate whether there's differences in antibody levels and the quality of the antibodies that are produced by immunizing with these two antigens. So when we looked at antibody levels, total IgG and sera, we found that there was no difference in the amount of antibodies that were elicited by these two different immunizing antigens. However, when we looked at the ability of these antibodies to block the interaction between TBPB and HDF, what we found was that by immunizing with a non-binding TBPB antigen, we were able to develop these mice were able to develop blocking antibodies that prevented TBPB from binding to human transferrin. So what we think is happening in this group over here is that in addition to other forms of immune-mediated protection, such as serum bactericidal effects, opsonal phagocytosis, and so on, these immunized animals are also able to block the bacteria from taking up transferrin, from interacting with transferrin and taking up iron. So in this group, we think that the accelerated clearance is also because of starvation of the bacteria by this, by the blocking antibodies. So while the HDF mouse model is the closest we can really get to modeling infection in humans at this point, we were actually very lucky that we had the opportunity to test our strategy in a native system. So TBPB and transferrin receptors are actually found in a variety of veterinary pathogens, and these transferrin receptors are specific for the transferrin of their host species. So we made use of a pig sepsis model of H. parasuis, which is a pathogen that affects pigs. And when we generated native and non-binding TBPB variants from H. parasuis and immunized those into pigs, we found that upon challenge, while the native TBPB antigen was 50% protective, the non-binding TBPB antigen conferred 100% protection. So this was, again, very promising and lended support to our hypothesis that a non-binding variant would be more protective in a native species. So in summary, today what I showed you is that the transferrin receptor proteins are promising targets for developing novel vaccines against multiple pathogens, and we think that we can utilize these receptor proteins to develop vaccines against gonorrhea, better vaccines against meningitis, and perhaps also otitis media. I've shown you that a strategy for engineering non-binding TBPB antigens was found to be superior to just utilizing the native TBPB antigen in the context of the native host. And we think that the way that this works is because the non-binding antigen is able to produce antibodies that are blocking the interaction between TBPB and transferrin, and therefore starving the bacteria upon infection. And while I focused more on TBPB in this talk, we really think that this strategy can be broadly applied to other protein antigens that have an interacting ligand in its host. So with that, I would like to thank everyone, my supervisors Scott and Trevor, and collaborators in Calgary and Brazil. Thank you.
Thank you all for coming back. And we're going to get started with the second half of our morning. This is a panel discussion on vaccines and immunity. And we're fortunate to have three speakers who will each do 10-minute presentations, and then we'll have a 15-minute panel discussion following. So I'll introduce them one by one. The first is Dr. Erin Wheeler, whose primary appointment is in the chemistry department at the University of Toronto, with cross appointments in the Donnelly Center and the Institute for Biomaterials and Biomedical Engineering. Dr. Wheeler is the Tier 1 Canada Research Chair in Microfluidic bioanalysis, and his research groups use microfluidics to solve problems in chemistry, biology, and medicine, and he's going to talk about portable diagnostics for serological surveillance of vaccine-preventable diseases. So I'll turn it over to Dr. Wheeler. Thank you.
Thank you so much for the introduction and the opportunity to participate in this very interesting and timely symposium. I'm asked first to make a disclosure. Unlike the previous speakers, I have some disclosures. I am a co-founder of a company that may eventually commercialize some of the techniques that we've been developing. I've also received some grant funding from Abbott. I've never done this before. I'm asked to explain how I'm going to address this in my presentation. I don't know. I mean, I trust you, dear audience, to make up your own mind. If this seems too biased, take a nap, and I'm sure we'll have less biased presentations later. Okay, so I'm here to talk about serological surveillance. And in particular, we're motivated by parts of the world in which we don't know the immunity status of our population. So particularly in remote populations and populations in areas with limited resources. Now, the critical thing to recognize here is that in these types of situations, we don't have enough vaccination dollars to vaccinate everybody all the time. That's the sad fact. So by knowing who is immune, who isn't, and how that's changing, we can make the best possible decision about when to spend those dollars. Unfortunately, the traditional techniques that we would use to make these decisions simply aren't tenable. But by the time we collect samples, we bring them back to the lab, we do the tests in the lab, and then return the information back to, for example, the refugee camp. By the time that happens, the population has completely changed. So there has to be another way to do this, and of course there are lots of researchers working in this area, but this is a place I've spent some time thinking about how to develop an on-site test to take into the field to get the answers immediately such that the information can be acted upon. We work in the area of microfluidics, and in particular we work with this technique that we call digital microfluidics. Typically the droplets aren't containing food coloring that's blue, pink, and yellow. Typically they don't spell U of T, but I think this serves as a nice example. So we've been working to adapt this tool to be able to run serological assays. Now, the assays and the chemistry themselves are not new, and in fact these are just standard ELISAs. What we have done and what we've spent a lot of time doing is figuring out how to make these assays work on the micro scale. So for example, we'll take a pinprick of blood, we'll inject a droplet of that pinprick of blood onto the device, we'll expose that pinprick of blood to, for example, a suspension of magnetic particles that will allow us to selectively pull out the analytes that we're looking for. And then in a series of additional steps that involves a lot of washing and a lot of additional reagents, we eventually get to the output, which is a product of a reaction that generates light that tells us that yes, the analyte was there, or no, it's not. I won't bore you with the details, but these are the kinds of things that my group and I spend a lot of time thinking about. Okay, so how do we make the devices? Well, one of the things that became clear to us is that they had to be inexpensive. We've spent a lot of time figuring out how to make inexpensive devices. This is a $100 inkjet printer in my group in which we're printing off a sheet of flexible devices. We've developed some inexpensive roll coating techniques. This is a technique that's sometimes used to apply paint to toys and other types of products, such that we can assemble the final device for about $1, which ends up being a reasonable price for making hundreds and thousands and taking into the field. The other thing we've spent a lot of time doing is figuring out how to build the instruments and how to build them so that they operate robustly and under conditions that we can't predict the environment. And you know, we don't have a production line somewhere doing this. This is us and our students making these things in the lab. This was an undergrad who worked with us a couple of years ago. This is in an office in the Donnelly Center, and you'll see it's about 12 hours time-lapse photography involved in making this instrument. This instrument was a little over $2,000. Our current instruments we're making are on the order of a few $100s. And each instrument then would work with lots and lots of the $1 cartridges. Okay, so a couple of years ago, we linked up with the US CDC and a long list of other partners, including the United Nations, and we decided to take our test out to a fairly difficult environment, the Kakuma refugee site, located in northwest Kenya. You can't just call Air Canada and get a ticket to this place. It's a charter flight operated by the United Nations once every week or so. And in the test, we evaluated pinprick blood samples on the spot from about 200 subjects. The same subjects, we also collected blood, spun down, froze, and sent the serum off to a central laboratory for testing. The rock curves are pretty good, area under curve 0.9, area under curve 0.86. We have some ideas about how to improve this going forward, but remember, this is a population who normally would not have access to any information, so we think this is a pretty good start. We have subsequently gone on a second field trial. This was to the DRC. We've heard a little bit about the DRC today. Thankfully, this was right before Ebola really started causing problems there. In this particular case, we weren't in a refugee site, and in fact, we were going with the CDC out to sites where there were suspected measles outbreaks, setting up shop, running the assays outside, and I'll point out that my student, Alex Clevanos, will present, I think, in just a little while to describe this test, so I won't spend much time on it. Okay, so finally I'll conclude by saying, is this only relevant in remote settings? And I wish the answer was yes. Of course, you know, I'm studying measles here. It's unfortunately, it's relevant here, right? So I pulled this story off the web 2 days ago. It's not just in Canada, it's all over the world that we're not vaccinating. And I hear a lot about hesitancy. Hesitancy sounds kind of mild. Sure, hesitancy, but I mean, according to the news, there are some forces working against us here. So if those of us on the side of vaccinating, we need to come up with some new strategies. I'm eager to be a part of this center. I'm eager to line up behind our fearless leader here. Folks like Shelley will come along and tell us all about the Toronto story. And I'm just happy to be a small part of it. With that, let me thank my research group, really outstanding group of folks who do all of this work. I'm just the guy who shows up to talk about it. I'll thank these organizations for funding. Thank you for your attention.
So our final speaker in our session on immunity is Dr. Tanya Watts. Dr. Watts is currently a professor of immunology at the University of Toronto and holds the Sanofi Pasteur Chair in Human Immunology. Her research is currently focused on understanding the role of T-cell lymphocytes in protective immunity of viruses, and she's going to speak to us about how to induce long-lived tissue-resident memory T-cells in influenza virus. Thank you, Tanya.
All right, thank you. Thank you to Natasha for all her work on this initiative, which I'm really happy to be a part of. So my lab's actually interested in basic immune mechanisms and how we can learn mechanisms of protection to improve vaccine design. So I do hold the Sanofi chair in human immunology. It's actually an arm's length chair, so I don't know if that's a conflict, but other than that, no conflict. So one of the models that we're very interested in our lab in studying immunity is influenza. And I probably don't need to tell this audience that seasonal influenza actually exerts a high toll, with the WHO estimating up to 500,000 deaths annually. And of course, we have an ongoing threat of pandemic influenza, such as happened in 2009 when there was a reassortment of viruses that led to a strain to which most of the world was not immune. And so the challenges in influenza are that we have this antigenic drift, which is mutations occurring all the time, minor mutations, and antigenic shifts, such as new pandemic strains. Another challenge that we've noticed is that there's really a short duration of protection of current vaccines, and even of immunity to influenza in circulation. So this is just a figure from a study we did with samples obtained during the 2009 pandemic, where we could take human PBMC and just use whole virus stimulation with the 2009 virus and ask how many T cells respond. And we actually happened to have one donor, a colleague, who'd actually taken his blood a year before the pandemic and then realized he had pandemic flu, got confirmed, and then took blood all the way through. And we even went back to him 2 years later. So we had the chance to ask, how does his T cell response emerge in the blood, the circulating T cells, over the course of the pandemic infection? And so from the onset of symptoms, his peak T cell response actually took almost 2 weeks to develop. So a little bit late to contain the infection, but enough so he wasn't hospitalized and would have been able to clear it. But what you can see, even this natural infection, actually the circulating T cell response by a couple of years later is more or less back to baseline. So he is immune, but it's really hard to get a really long-lived high level of immunity that would be enough to immediately protect with T cells. So what are the approaches people are thinking about for vaccines? So I work on the T cell side, but right now there's a lot of interest in improving antibody responses to flu. Most of you probably know that the trivalent inactivated vaccine largely induces antibodies that bind around the receptor binding site to prevent infection, so neutralizing antibodies. But this puts a lot of selective pressure on the virus, which can escape, and soon your vaccine protection disappears. But people have discovered that there's a region of the hemagglutinin, the fusion peptide of the stem, that can't change, otherwise the virus loses infectivity. And so there's a lot of interest in inducing stem-reactive antibodies. And even more recently, there's been another conserved region found at the trimer interface of the hemagglutinin. And interestingly, those antibodies are probably not working by neutralization, but by causing death of infected cells. So actually, the NIH Vaccine Research Center just started a trial for antibodies based on this epitope in the hemagglutinin stem. However, I'd like to point out that T cell responses are also very important. So the reason T cell responses are important is several-fold, but first of all, T cells recognize conserved regions of the influenza. So we can show that the very old strains like influenza 1934 cross-reacts with the 2009 strain. And also, during a primary immune response, in order to get a very long-lived antibody response, those B cells actually need help from T cells in order to become long-lived memory cells, as well as long-lived antibody-producing plasma cells. And then the other thing, of course, is if you don't have an absolutely perfect protection with your antibodies, you need the cytotoxic T cells to kill the infected epithelium and get rid of the infection. So my lab really studies T cell immunity to viruses and really trying to understand what it is that controls their response. So the initial response happens when a T cell sees the virus presented by a dendritic cell in the lymph node. You get this rapid expansion of a clone of T cells that again goes out to the site of infection and clears the virus. And then some of those cells persist as memory cells. And there's a lot of interest in understanding how do you get a large and long-lived memory response. And also a question of ours is how does the immune system know how long to keep this T cell response going in the tissue? So when the T cells get to the respiratory tract, you want them to stay around to clear the infection, but not so long that you get pathology. And so we study a family of receptors in the tumor necrosis factor receptor superfamily. Don't need to worry about the details, but we've shown particularly for a couple of these family members, they're induced on the T cells during infection, and they recognize their ligands not only in the lymph node, but again in the tissues while the virus persists, and this gives them a survival signal that is really tuned to the duration of infection. So a new concept in the last 10 years that's come out in the immune system is that we not only have these circulating cells that we measure in the blood, But there's a population of T cells called tissue-resident memory cells that actually form, they differentiate locally in situ during an infection, shown here, for example, in the lung, but in the skin, mucosal tissues, depending on the site of infection, and respond to local cues from infection to become a non-circulating population. And if you deplete all the circulating cells, these cells are sufficient to protect against influenza. And because they don't recirculate, we actually don't measure them in the blood when we're looking at correlates of protection. So this is something we can study quite easily in mice and much more difficult in human. And it's been shown that they really form this first line of defense against reinfection, rapidly producing cytokines to attract the other cells to come back and initially controlling it. So we're really interested in how can we design an experimental approach to enhance tissue-resident memory and ultimately inform vaccine design. And also, I think a lot of people in the field are interested in what you could measure in the blood that would be a correlate of having good memory in the tissues, which is where you really need the protection. So I'm not going to give you much detail, but in the last couple of years, what we've found is that we've known for a long time that the initial activation of T cells in the lymph node actually requires 3 signals: the antigen-specific signal, co-stimulatory signals, and cytokine signals that together activate the T cells and allow them to expand and differentiate appropriately for the kind of infection. More recently, we found that during an infection, another population of cells, the inflammatory monocyte-derived antigen-presenting cells that are hardly present at all in the uninfected tissue, are very important in providing another signal we're calling signal 4, the TNF superfamily ligands, which actually happens in the lymph node, but even more dramatically in the lung during flu infection, to give this survival or go signal to the T cells when they get to the tissue. And we've also found that this is very important for actually the development of this tissue-resident memory population. So just to give you one example, without going into details, we can engineer a mouse so that the bone marrow-derived cells actually half come from a wild type and half for a knockout in this family of receptors on T cells. And you can show that if you look in the lung tissue, that the cells that have this signal vastly outnumber the cells that lack the signal. And this is true both at day 9, the peak of the viral response to influenza as the T cell response, and also when you go later and look at these tissue-resident memory population. So this told us that this was actually very important signaling to get tissue-resident memory. And so we've actually tested a way of kind of getting more tissue-resident memory. And from earlier work, we knew that actually this protocol worked very much better as a boost protocol, where we gave a mouse influenza and then waited a month to mimic somebody who's already influenza immune. And then we used a replication defective adenovirus with either the influenza nucleoprotein or expressing the nucleoprotein plus one of the ligands of this family that boosts T cell memory. And then we waited about a year, so we actually had quite old mice by that time. We finally had to stop because some of them were dying of old age. We didn't want to lose the long experiment. And what we found was that there was a remarkable, so this was the T cell, CD8 T cell response to the initial infection. And with this co-stimulatory molecule, we could vastly boost this circulating population in the mouse blood and amazingly get a very long persistent protection. And then when we looked at the lung resident cells at different time points, you saw that we could really enhance this, but importantly, this could actually persist out to, here we measured at 200 days, so that we were getting this establishment of a long-lived memory population. And then when we challenged 2 cohorts of mice, you could see they were protected from weight loss and from death if they had these dual signals. Importantly, when you look at the route of delivery of these vaccines, if you give it systemically by an intraperitoneal injection versus intranasally, you get a very different result in the lung tissue. So in the spleen, the systemic vaccination gives you a really nice big boost in the T cell response with this dual molecule expressing vector. But in the lung, you really needed the intranasal infection and not the systemic infection to get this lung resident population. So it's really important. Am I going over? Okay, I'll just skip to the end. And it was also important to deliver the two cells locally in the lung. So basically, my take-home message is that from what we now know about tissue-resident memory, the signals that are delivered locally to the lung are very important in establishing a protective response in the local tissue. And we're working, rather than using a vaccine with a self-molecule, in figuring out the kind of signals, the specific adjuvants that can give us this really strong response locally. So I'd just like to end by thanking the people in the lab who did this work. These two particularly are working on the tissue-resident memory, and also William Chu. And thank you to our funders.
Thank you very much, Dr. Watts. So 3 very interesting presentations that deal with the immunity related to vaccines and disease. And so I'd like to call up the 3 speakers, Dr. Bolton, Dr. Watts, and Dr. Wheeler, and open up for questions. We have about 7 or 8 minutes for questions. So questions from the room around any of the 3 topics. Yep, go ahead.
All right, thank you very much for your presentations. Oh, sorry.
Oh, sorry. Lawrence, go ahead, and then we'll take the speaker in front.
Apologies. Thanks very much for your presentation. So, Shelley, this is actually a question just around the numbers from Brazil. And I'm curious if there are, given that there are many other jurisdictions that have eliminated but have not seen such a dramatic rise, are there any unique factors that led to what the picture we're seeing in Brazil now?
So I think that a very unique factor is migration from out of Venezuela. That is a migrating population without access to vaccines. But I think that the bottom line is that if those particular states had adequate coverage, then this might not have happened. And I'm wondering, actually, Natasha, if you have anything to add from the PAHO perspective?
And just to repeat the question, that was about why we're seeing the resurgence in Brazil, right? Yeah.
Yeah, so I sit on the committee for the Pan American Health Organization that's looking at this at the moment. And I mean, you're right, Shelley, the origin of it all was the political breakdown in Venezuela that led to the disruption of the immunization program there, and also the exploitation of people. So that was like the starting point, but it's a bit like this forest fire thing where, you know, somebody can set a forest fire and that's a bad thing, but it doesn't take off unless it meets a dry forest and there aren't the right controls in place. So, you know, Brazil had a gap in its immunization coverage. And it's a bit like Canada. It's got very decentralized federal state, so we have a lot of lessons to learn. And the outbreak response is organized at local level. And so and it's much more complex to get, and it's a big country, I mean, it's a much bigger population than Canada, but it's complex to get stronger immunization programs because you're working with multiple levels of authority. And then thrown into the mix as well is in the Amazon region, there's a big indigenous community, again, like echoes of Canada, that are very mistrustful of authority and who were not getting access to immunization. And so there are a lot of ingredients that went into that Brazilian outbreak, and they're a very well-organized, smart nation that, you know, are dealing with it. But as I said, it's that forest fire analogy I think works quite well for measles because it takes off so quickly. And we have the same issues in Canada, where there are parts of Canada where we are, you know, storing up trouble if we don't get ahead of it. And I think that's one of the interesting things about CIREPI is it can tell us not just where measles has come from, but where it might go next. And that, I think, is one of the strengths of that approach, telling you where our gaps might be and where we need to jump on them and try and fill them. Thanks.
Thank you. So there was a gentleman right behind, and then I'll come to you as well. So the question was with just regard to the decay of vaccination titers over time and how that relates to other vaccines. So Tanya, do you want to start?
Yeah, I mean, I think this is a huge question in the field. So if you look at studies of vaccinia responses, so smallpox 30 years after elimination, you have the antibody response, this is work from Rafi Ahmed at the Emory Vaccine Center, was almost undiminished, and the T-cell response was extremely long-lived. He recently did a study of yellow fever vaccination in people who didn't get any further exposure. They were in a non-endemic region, and the T-cell half-life is 473 days. So those are two examples, smallpox and yellow fever, of live attenuated viruses that give lifelong immunity. But yet we have other examples like measles, which is a live attenuated virus that doesn't seem to last as well. And so to me, I don't think we know the molecular insights yet, and I really think we need a lot of research and to understand. There's a lot of activity in the field about subsets of T cells and what happens in the tissue, and we really need to figure out those cues and the differences between those models.
So just to add, just going back to that slide, there are many different things like the host, the vaccine, the setting around them, like it's the triad of host vaccine or host pathogen environment. We are following up with a study that we were just recently funded for, where we're going to look at the sera of people who have waned, and we're able to link their information to administrative data to really get in at the population level to see what their risk factors are. And until we do that, it's all very circumstantial, and it makes a pretty picture, but you can't say anything for sure. So we'll be able to dig into vaccine status, place of birth, disease history, socioeconomic status to really unpack it. So the beauty, and thank you for letting me plug my my research program. The beauty of the platform is it's actually an MMRV platform. It's a measles, mumps, rubella, varicella platform. So once we've done the testing, we're able to build blocks on top of it.
Just a reminder that we do have people online, and so if anyone is interested online in asking a question, please feel free to type it in the chat box. So I see three questions, 1, 2, 3. Go ahead.
Hi, this is for Dr.
Sorry, sorry, in the front, and then I'll take you in the back as well. So sorry, I'm not very specific in my pointing. We'll take you as well. Go ahead. So if you just repeat.
So the question was about whether we have parameterized mathematical models with our sera epi data, and the answer is that is in progress. We're collaborating with Marc Brisson out of Université Laval, and he will take our data. I think with the pilot data of 1,200 sera, we'll see kind of how far we can get, but we are getting larger data sets that are national as well as follow-up data. We have done some mixture modeling already for varicella and also for measles. It's a vaccinated population and it's presenting some challenges. So unlike a population that's either really immune because they've had measles or really susceptible, we have susceptible, naturally immune, 1 dose, 2 dose, waned and boosted. So we'll see how it pans out.
Okay, I'll get the microphone and then we'll come up front.
Hi, it's a question for Dr. Shelley again. Just to that small population you were talking about with maternal risk factors, I'm just curious whether the breastfeeding status of the infants was looked at all? That's all.
So the data that I presented was pilot data, and it was done in partnership with Michelle's Science at SickKids, and Michelle went through all of the EMR, all the electronic medical records, and as you can imagine, there's not much information on breastfeeding, but we're now doing a larger study where we're recruiting prospectively, and we're collecting samples and data from mother and from baby, including breast milk. And so that is very exciting because I'm personally really interested in the contribution of breast milk to immunity. But again, it's not going to be straightforward because breast milk is different types of antibodies. There's no correlate of protection, and in fact, the assay doesn't exist yet to test it. So we're like many years away.
So the question, just for people online, was about with healthcare workers, we currently say that you're immune if you've had either serological demonstration or 2 doses of vaccine. With waning immunity, how does that impact precautions that healthcare workers should use, given that they may not all be immune just by those parameters?
So I think that's a really good question. We have data from other jurisdictions, like for example, in the Netherlands, there was a hospital outbreak that included, I believe, 8 healthcare workers, 5 of whom had been previously vaccinated, and because they were healthcare workers, their sera were banked, and 2, I believe it's 2 out of the 5, should have been immune because they had just done sera. So we're certainly seeing things that we wouldn't have expected. We also see evidence of vaccinated individuals being infected and then transmitting to other people. So, I mean, that's all very scary, but I think we would need to look at it systematically. We're in the process of doing that more generally, not specific for healthcare workers. In terms of actual evidence that's based on stuff that's already happened as opposed to our perception of risk, there is, but we haven't looked at it systematically.
So there was a question at the back. Yep, go ahead. So the question was related to the cost of, sorry, the cost of immunity? Okay, the cost of laboratory tests for testing for immunity. We have 2 candidate responses here.
Well, because there's different methods of doing it, so Aaron, why don't you go first? I think Aaron's cost is better, so.
Well, I mean, but my cost is very experimental, but I mean, using the devices we make in our lab, we're looking at cents for a test, yeah.
Okay, and then using our platform, we have an enzyme immunoassay that's a commercial enzyme immunoassay, and it's a quadruplex, and we get it at $3 a target, but that's an absolute deal, that's not retail. So for someone who has natural immunity, that would be enough, because then you'd have off-the-chart ELISA results and we wouldn't need to go further. But if we need to reference test, we do that at the National Microbiology Lab. And again, not retail, it's about $20 per sample plus tech time.
I just wanted to make a comment too. I mean, I guess a question for Shelley, which is, are our correlates of protection, which is an antibody ELISA, really accurately reflecting protection? And is it really a failure in immunity due to us following the wrong parameter?
So the answer is, in my opinion, no, they are not. We did this as part of our systematic review, and again, reviewed 15,000 papers, came up with 5 that were pertinent. And the correlative protection of 120 MIU per mil was based on an outbreak at BU, at Boston University, where there was a blood drive at a dorm, and then there was a measles outbreak. So it's this opportunity that doesn't come along very often, but it was based on an N of 8. So certainly we're seeing, you know, it's not all about humoral immunity, as you know better than anyone, first of all, and second of all, as the immunity of populations shift, maybe the correlate needs to shift too.
So I am cognizant of the time, and I know there are a few remaining questions, so if I can encourage the few remaining people to approach our speakers after that would be great. I just don't want to hold us up too late. But if you could join me in thanking our 3 speakers with very interesting talks on immunity. Okay. Okay, so now we're heading into our final part of our morning agenda prior to lunch, and this is the first of two parts which have been a tradition in our Vaccine Sciences Symposium, which are our student presentations. So we're going to have 3 student presentations this morning and 3 student presentations this afternoon, and I'd like to start by introducing our famous judges. So if you could just stand up and wave. We have 3 judges from Public Health Ontario, Dr. Julian Koos, who is over there, and Dr. Sarah Buchan over there, and Dr. Shelley Bulletin, who you just heard from before, over there. So those are, you might want to direct your presentations in that direction. So we will have 3 speakers, and I'll introduce them in sequence, and then we'll have a time for a few minutes afterwards for some questions to our student speakers. So our first speaker is Dr. Elisa Curry, who's a PhD student in molecular genetics at the University of Toronto under the mentorship of Dr. Scott Gray Owen. And Elisa will be talking to us about revealing processes that confer mucosal immunity against Neisseria meningitidis. So, Lisa, thanks. Come on up.
Hi, everyone. So just to start, I have no disclosures to address. So my project deals with the bacterial pathogen Neisseria meningitidis. And so this bacteria is a major cause of sepsis and meningitis worldwide. And of these infections, fatality rates can be upwards of 10%, making it a very important vaccine preventable disease. However, in addition to this invasive disease, Neisseria meningitidis is a regular colonizer of the human nasopharynx. While in the nose, this bacteria causes no harm to its host, however, it can be easily transmitted throughout the population. And so when we think about designing a vaccine against this bacteria, we know that an effective vaccine must be able to prevent not only invasive disease, but also nasal colonization. Unfortunately, we don't really understand the immune processes that confer protection against nasal colonization, and this was recently evidenced in which novel vaccines that were brought into the human population actually failed to have any impact on nasal colonization rates. And so this is where my project comes in which I want to identify what immune cells are associated with protection against nasal colonization. In order to do this, I make use of a mouse model of nasal colonization, in which I vaccinate mice with a vaccine previously shown to prevent nasal colonization, and then before I infect the mice, I deplete them of a target immune cell. The thinking here is that if the immune cell is required for protection, the depletion should render the mouse susceptible, and therefore when I infect them, I should recover bacteria from their nose. In contrast, if the immune cell is not required, following depletion, the mouse should remain protected, and I should recover no bacteria from their noses. So following this methodology, I have discovered that both B cells and neutrophils are required at the time of infection to protect mice against nasal colonization, while T cells in the complement system appear to be dispensable. This is particularly exciting in the context of neutrophils because while they are known to be very important in killing bacteria, they've never been associated with vaccine-directed immunity against this particular bacterial pathogen before. So it is my hope that with this information, we can better design vaccines against this bacterial pathogen, potentially that specifically target the activation and recruitment of B cells and neutrophils to the nose and reduce infections worldwide. And thank you.
Thank you very much, Elisa. So our second presentation is Vinyas Harish, who is an MD PhD candidate at the University of Toronto Faculty of Medicine and the Dalla Lana School of Public Health, working under the mentorship of Dr. Cameron Kahn. So Vinyas is going to talk about an air travel analysis of the 2017-2018 mumps outbreak in Hawaii.
Perfect. Hello, everyone. My name is Vinyas, and I am a second-year MD PhD student at the University of Toronto. In terms of, in terms of my disclosure, the work I'm about to present was funded summer studentship at Dr. Khan's company, Blue Dot. But to mitigate that, the data sources that I've used in this study will be discussed, as well as our methods. So imagine that you and your loved ones are taking a trip to Hawaii. Whether it's the pristine beaches or the rolling hills and mountains, it sure seems like the trip of a lifetime. But what would happen if you saw those news articles? Hawaii mumps outbreak stumps experts. Hawaii typically sees 10 cases of mumps a year. An outbreak has now sickened 100 times that. And how would you feel if someone in your family was immunocompromised or wasn't vaccinated? We know mumps can be dangerous and lead to complications such as orchitis, deafness, meningitis, and more. So I like to think that this is a question that many people are thinking about, especially because the 2017-2018 mumps outbreak had 1,009 cases. So the question myself and other summer students at Blue Dot this summer wanted to think about was, what was the risk of exportation of mumps, both domestically and internationally, given that Hawaii is a major international travel destination with 9.4 million visitors in the last year? To answer this question, we used the International Air Travel Association's database. And we can break the analysis down domestically and internationally. So we use the IATA travel database, as well as the CDC's Teen Vax NIS Teen Survey survey from 2016, and found that several states with high amounts of travel volume have coverage estimates that may be less than the threshold needed for herd immunity, and contemporary estimates around 88 to 92% coverage needed. And this doesn't even take into account what we talked about today with patterns of waning immunity or subpopulations in these states who may be more susceptible. Internationally, we see similar trends. Notably, Japan, with 50% of all outgoing travel volume, only has 30 to 40% estimated mumps coverage, and that's because mumps isn't in its national vaccine program. Canada, Korea, and China are also estimated to have lower coverage than necessary. Australia is doing pretty good, though. Ultimately, what this study shows is, although this outbreak did end in October, the risk of exportation could have been significant, and really goes to show the importance of promoting international vaccine schedules. Thank you.
Thank you, Vinis. So our final student presentation for the morning is Alexandros Sklavounos, who is a PhD student in chemistry at the University of Toronto under the mentorship of Dr. Aaron Wheeler. So Alexandra, I'll pass it over to you to talk to us about a portable lab design for serosurveys and seroprevalence at the point of care that we heard a bit about this morning.
Thank you very much. And thank you for inviting me here today. My supervisor gave a great introduction to what we're practically doing in the lab in his talk. At this point, I have to say that, I have nothing to disclose. And let's start with what the real issue is. So what we're doing here, we're fighting against the vaccine-preventable diseases. And there have been collective efforts focused on eliminating these diseases, and especially measles and rubella the past years. But unfortunately, we're not there yet. And as Dr. Shelley stressed before, instead of going forward, we're going backwards. I'm not gonna talk about why is that happening, but one of the reasons behind that is the serological tests. And why do we need those tests? Because during the epidemiological studies, the ones that actually drive the response to the epidemics, there is a huge bottleneck between the sample collection and the actual analysis and the results. And that bottleneck is the transport to the facility as well as the analysis itself. It takes so long to go from the sample to the result. So our question here is, how can we expedite this process? And our solution is what we call as MRBox, and it's version 2, because as my supervisor showed before, we had another box, the version 1, which went to Kenya. Our box relies on a technology that we call as digital microfluidics. And digital microfluidics is a fluid handling technique that allows us to move, dispense, merge, and split droplets in an automated fashion. We have also developed, apart from the hardware, a software that allows us to perform all these processes in a completely automated way. So we have a box that can do the fluid handling completely automated. It includes a magnet to perform complicated immunoassays on beads, which will capture the analyte and will help us to move to the place where we will receive our readout. It is battery powered, so we can take it anywhere. And of course, we decided to put a very sensitive sensor that will collect light. So we're using a reaction that actually produces light. And we want to collect that tiny bit of light just with this detector. So we're doing what we call as chemiluminescence. One other thing that we did in the lab is we actually adapted and developed assays that will take whole blood and will turn it into a result in under an hour. And what we're doing here is we're not competing against those assays that will give us a yes or no result, but we have actual quantitative data from our box. So we are at the level of a laboratory test, but we can take it anywhere. And when I mean anywhere, it can be from Northern Ontario up to any other place. And what we decided to do is to take our box to the real test. So we took it to the Democratic Republic of Congo, where we partnered with the CDC to run 2 different studies. The one was the zero survey study, where we only looked at the samples from children and women at childbearing age, while we also collected samples for an investigation study. And in total, we collected 357 blood samples that we ran in only 24 days. So in 24 days, we had the sample and the result right there, while we were still waiting for the gold standard for many, many months later on. So thank you very much for this.
Thank you very much, Alexandra. So 3 very interesting and very excellent talks, and the students are now available for questions about any of their research. Tanya, go ahead. So the question was how long neutrophils are short-lived, and so how long after vaccine did you challenge? And the second part was? Really, is there a mechanism where a long-lived part of the immune system is boosting the neutrophils at the time of challenge? And is there a long-lived part of the vaccine immune system that's boosting at the time of challenge? You can tackle that one.
Thank you. Good question. Yeah, neutrophils have a very short life, especially systemically. And so when I vaccinated, the mice were challenged 2 weeks after their second dose of the vaccine. And so what I think is really happening is that some part of the adaptive immune system, whether it be T cells or B cells, is increasing recruitment of the neutrophils to the site of infection when I infect. And that is accounting for the short-lived aspect. Thank you.
Thanks. Yes, go ahead. Oh, sorry. Go ahead in front and then in back. So the question was if there's any difference in the immunologic response to polysaccharide vaccine from meningococcal?
So my focus has been on the polysaccharide vaccine, which, for those unfamiliar, the polysaccharide vaccine has been implemented since the early 2000s and is very good at preventing nasal colonization. And so I haven't done a lot of work on the protein vaccines yet because we wanted to set our basis of what works before going into the protein vaccines, which are new. So we'll see.
Yep, go ahead, Lawrence.
Perfect. So for the presenter on the Hawaii flight question, so I noticed you used coverage rates as well as volume between the places. I was wondering if you looked at flight frequencies at all, because I think for that reason, it was like we know that what happens, for example, in Manhattan is much more likely to happen here in Toronto, just given how frequent and regular flights and people are being introduced between the two cities, as opposed to somewhere like, I'm trying to pick a place like St. Louis, where I think we only have like 1 or 2 daily flights. So frequencies of flight, was that something that you also maybe considered adding to your model? Because I think that'd be really interesting.
So the question for Vinyas about how flight frequencies might have been incorporated into the model.
Thank you for a great question. The answer in our analysis was no, but Blue Dot is working on developing models that does incorporate that, and they've got some pretty exciting partnerships with airlines. Like Air Canada was something that was announced pretty frequently, or pretty recently, and that's something that they're looking at for future analyses, but for the purposes of our study, no.
Great work. Very, very curious to see the future. Thanks.
Thanks so much.
Yep. Sorry. So we'll start with Tanya, and then we'll go Natasha, and then we'll go to the back. Oh, go to the back. Okay, so Tanya and then the back. Okay. Can you repeat the question for the audience?
Yes. So if we can actually move further away from just the immunoassays and run other assays. So the good thing with our platform is that it is generic and it can be paired with different detectors, different assay schemes. So this is just an example of what our box can do. Yes, it can do immunoassays, but we can always do other type of assays like PCR or an isothermal amplification. We're actually working on something like this with the Party Lab, where we are developing a Zika diagnostic platform, and we will probably deploy a new version of the box in the field sometime soon. And yes, the sky is the limit for that, so I'm very confident about that. Yeah. Yes.
I'd like to ask Dr. Simonis, what do you expect the price to be when it goes retail?
That's a great question. And we, when we first develop our tips and our assays in the lab, we don't actually have the price in our mind, but when we come to the point where we want to publish it or even commercialize it, we have to face that. So in the lab, the test may cost $10 or more per test, but we strive to bring that cost down. And as my supervisor mentioned before, right now our chips cost about less than $1 actually to produce. And that's actually for a small scale. So even if we up the scale, we can always bring the cost of our systems lower. The other thing is that because we're using such a small amount of reagents, we actually don't need that much. So most of the cost of these assays is the reagent. And because we use such a tiny amount, the cost of our system will also decrease. Thank you. So the hardware is something that the company, our spin-off from the lab, but my supervisor can also like follow up with this.
Buy a research version for $5,000, but a dedicated instrument is going to be much, much cheaper.
Yeah. Thank you.
Just for on the line, the cost of the box for research version was $5,000, and other versions would be cheaper? Okay. Natasha, go ahead.
Thanks very much. It's the same speaker again, so thank you. I just wondered, given what we'd heard from Dr. Bolton earlier, what lessons you had from Democratic Republic of Congo you might apply to rolling out a point of care test in Canada, because it sounds like, especially with that costing discussion, it might be a lot cheaper to use your technology in Canada than what we have at the moment.
Yeah, this is also a great question, and if we can actually take our platform is not just for the Congo, so let's start with the first part of the question, which was what actually we got from our trip there. Is that correct? This is what you were asking. So we did see that performing at the point of care, it doesn't only speed the things up, but just for the point that time, we were just doing only the epidemiological study, but if we could partner with other sources like UHN, or we can actually do the vaccination right there, we can always run the test and right after vaccinate the people who are in need of vaccination. So our platform can very nicely be paired with this type of campaigns. Now, when it comes to taking it to a different place, I believe that it would be great to be deployed in places like Ontario, and especially in places that is further away, where people don't have access to centralized hospital facilities. I think our platform would be great for that.
Thank you.
You're welcome.
Okay, any final questions or any questions online? Yeah, go ahead. So the question was about the 300 samples that were collected and the time interval it took to get the results?
Thank you for your question. So that actually has two parts. The first part, it has to do with what we decided that we will be using for our platform. So right now on our platform, we can load 3 samples, 3 whole blood samples and 2 controls, because we decided to take that hit of the running more samples by controlling our system and having standards to make sure that our results would be as good as the lab's result. So we can run 3 samples, but we can always expand our device to take more. So this is the one side. The other one is that we didn't collect 350 samples in one day. So samples were collected every day. There were approximately 10 to 15 samples collected every day, even more depending on its type of the analysis, because just collecting the samples for the surveillance is relatively easy, but the suspected cases, you actually need to have an idea and know that a person is a suspected case for either measles or rubella, right? So that cannot happen in a day or 2. So that definitely takes some time. I hope that I answered your question. Thank you.
Okay, so if there's no other questions, I'd just like to have you join me in thanking the 3 students for their excellent presentations. And that concludes the morning part of our session. So we do have a break from now till 1, and lunch is in the back and the room on the left. And Thank you very much for your attention, and we'll see you back at 1 o'clock.
And before we start, just to say, if any of you have arrived and for one reason or another didn't manage to pick up your name tag or tell people you were here, could you just let us know either at some point in the break, go and get a name tag? Not because especially if you want to have CME credits, because we need to know who's here so we can send out the right messages and get the evaluation to you. So if you haven't, it's just a few of you might have just come in without picking up a name tag. So welcome back, everyone. I hope you enjoyed your lunch. The afternoon is going to be chaired by Dr. Shelley Deeks, and I'm absolutely delighted to have her sharing the afternoon. I mentioned earlier, probably 10 years ago, we had a plan to try and get a Centre of Excellence in Ontario. And if I remember correctly, the name of it was the Ontario Immunisation Centre of Excellence, which spelled out on ice, which is exactly what happened. So it's great to be now speaking with it off ice. So Shelley Deeks is the Chief of Communicable Diseases, Emergency Preparedness and Response at Public Health Ontario. She's an Associate Professor at the Dalla Lana School of Public Health at the University of Toronto. She's Vice Chair of Canada's National Advisory Committee on Immunisation. And Dr. Deeks holds fellowships in public health in both Canada and Australia. She's over 20 years experience as a public health physician and has worked at all levels in the public health system in Canada and at the national level in Australia. So welcome, Shelley, and thank you for chairing the afternoon.
Thanks so much, Natasha. And I heard I missed a fabulous afternoon. I'm one of those guilty parties that came without picking up my name tag, so I'll do that at the break. But the next part of the session today is another round of student presentations. So I'm going to introduce the three students and then have them come up and present, and then we'll have a discussion with all three. So the first student will be Melissa Perry, and Melissa is an MPH student in social and behavioral science at the Dalla Lana School of Public Health, working under the mentorship of Dr. Charlotte Lombardo. The second is Kate Allen. Kate is a PhD student in social work at the University of Toronto under the mentorship of Dr. Shelley Craig and Barbara Fallon. And the third is Corinne Moss, and Corinne is an undergraduate in life sciences at McMaster University. She conducted research she is presenting under the mentorship of Dr. Anna Taddeo, who's here at the University of Toronto School of Pharmacy. So can I have Melissa, Kate, and Corinne come up for your presentations? And first up is Melissa.
Good afternoon, everyone. I hope you've enjoyed your morning so far. I'm going to kick off the afternoon speaking to some findings from an evaluation that was done on HPV vaccine information on public health unit websites in Ontario. So I have no conflicts of interest to present so far. So picture this. Your child, who's in grade 7, is due for their first dose of the HPV vaccine. You don't have the best relationship with your family doctor, and you're overwhelmed with the controversy surrounding vaccines. So you branch out and you look to your local public health unit website for some information. You become incredibly frustrated with the lack of available HPV vaccine information, and you fail to see the urgency and necessity of the vaccine. So what do? The 2017-2018 school year coverage rates for HPV vaccination in Ontario were 59.9%, and although represents an improvement from the 2016-2017 school year, are still substantially below the national target rate of 90%. When comparing HPV vaccinations to other school-based vaccination programs, both initiation and completion are much lower. Many parents are now using the internet for pediatric health information related to vaccines, which has been shown to lead to vaccine hesitancy and non-medical exemptions for children. This growing issue exemplifies the need for publicly accessible information surrounding vaccine efficacy and safety. It is essential to improve the information on public health unit websites, which are meant to be a key resource for Ontarians. Due to the discrepancies in HPV vaccination rates in Ontario, and the increasingly evident link between health communication and vaccine initiation, my co-author Julian and myself conducted a scan of the 36 Public Health Unit websites' information on HPV vaccine. In order to assess the consistency and quality of the information, the Communication Assessment Checklist in Health, known as CATCH, was utilized. This 12-step comprehensive checklist, known to assess health information, aided in the analysis of factors including layout, scientific evidence, risk communication, and language utilized. It was found that only 69% of websites spoke to what HPV was, 50% spoke to prevention measures, 44% spoke to side effects, and 28% provided misleading information. A cross-reference scan was also conducted that demonstrated a correlation between high CATCH scores and high vaccination rates, and low CATCH scores and low vaccination rates. After conducting this analysis, it was found that strengths include the comprehensiveness of the tool and the base that this provides for future researchers. Limitations include the potential bias of the CATCH tool and the fact that it was created for paper material. As discussed, HPV vaccination disparities within Ontario demonstrate the need for public health units to provide effective and consistent information for current and future public health of Ontario. Thank you.
And next we have Kate Allen. Welcome, Kate.
Hi there. Whoops, I'll put this up a little higher. So just to give you a little bit of background on our study, 2 years ago, my colleagues and I actually sent out a monthly mailing to pediatricians across Canada asking about what their experience with vaccine hesitancy was. So how often they encountered it, what kind of concerns parents brought up, and what the impact on their practice was. And we found that not surprisingly, it was ubiquitous. Almost 90% of our sample had come across it, and it really did impact their practice as far as time, resources, and the rapport with their patients and parents. And so that led us to want to understand what happens when a child actually contracts a vaccine-preventable disease. So what does that situation look like? How do parents respond in those scenarios, and how do physicians respond as well? So that's the crux of what I'll be talking about today. I have no conflicts of interest to declare, and same with my co-authors. So in terms of our survey respondents, the Canadian Pediatric Society sends out monthly mailings, and we did our survey as part of that. So they have a large membership that includes pediatricians and pediatric subspecialists. And we found that 21% of those who responded to our survey had encountered a patient with a vaccine-preventable illness in the last 12 months who had been intentionally unvaccinated or partially vaccinated by their parent. So it was about 1 in 5, which actually lines up with what we know about incidences of vaccine-preventable diseases in our population in Canada, so it's probably not an overestimate based on who answered the survey. As far as the types of VPDs that they came across, pertussis, pneumococcal vaccine, and varicella were the most common, and that actually also lines up with what we know in terms of incidences of these diseases across Canada. These are all considered moderate incidence vaccine-preventable diseases, so that does line up. And just to note, most of our sample was quite young, so the majority, about 90%, was under the age of 8, and about 25% of our sample was actually under the age of 1. So this was a very vulnerable group that would be very subject to a lot of complications associated with these illnesses. Oh, and also to note, these 3 vaccines, also the vaccine coverage for them falls below our national targets. As far as what the concerns were, what was interesting about this is actually we found that some of the concerns that parents had were very similar to the ones that were mentioned in the first study that we did. So the parents that weren't already not vaccinating their child, but that had concerns that they brought up to their pediatrician. So the main ones were the risk of the vaccine being greater than the disease, having too many vaccines in the schedule, that it would weaken the immune system, or that there were vaccine additives that they were concerned about. So what's interesting actually is in the subsequent survey, autism wasn't actually in the top 4, so I don't know if it's one of those things that we're moving further away from the Wakefield study, it's less top of mind, or it was sort of included in the risk of vaccine being greater than the disease, that sort of piece of it. So this was, I think, the most interesting piece of our study. And this is, we asked the pediatrician respondents how likely they felt it was that these parents would go on to vaccinate their child or the child's sibling following the vaccine preventable illness. So obviously against other diseases that they didn't actually just contract. And we actually found that it was about 1/3 of our sample. So this was very surprising that they felt confident that the parent would vaccinate, because we know that it works the other way. So we know that when there's a lot of media publicity around adverse reactions to vaccination, definitely cross-culturally we know this, there is more of an enhancement of vaccine hesitancy. More people have concerns. So it stands to reason that we would think it would work the other way as well. When there would be a parent who would be witnessing their own child with a vaccine preventable disease, that we would think that would maybe motivate them more towards vaccine uptake, but that wasn't really what we found. And what's interesting to note too is that we asked about severity of the child's treatment. So most of these children, as I mentioned, who were quite young, were treated in emergency departments, in inpatients, in hospitals, in ICUs, and in some very rare and unfortunate cases, there were child fatalities as well. So these weren't children that were mildly ill, these were children who were quite ill. So just to sort of summarize what we have here, we know from the literature that health professionals, and particularly the general pediatrician who sees so many families, is a really key component of vaccine uptake. We know that from the literature. But the problem is we don't really know what the best technique to be speaking to parents about this is. We know that participatory approach versus we know that a presumptive approach versus a participatory approach is better. So saying, we're going to do some vaccinations today as opposed to, what do you want to do about vaccines? We know that's more effective. And we have some ideas from the rest of the health literature that motivational interviewing might be something that holds promise. But there really hasn't been a lot of rigorous evaluation of what approaches there can be to deal with this issue. So I think that's sort of where our future lies as far as research, really understanding that for these key players, especially general practitioners and pediatricians that have this role with parents, how can we be advising them as to how to communicate around vaccination, not only for children who haven't yet been vaccinated, but for children who've contracted a vaccine preventable illness, to prevent further harm and illness for them as well. So hopefully I haven't left you with more questions than answers, but I very much appreciate your attention, and I'm happy to take questions, I guess, after all of them. So thank you very much.
And our final presenter in the series is Corinne Moss. Welcome, Corinne.
Good afternoon, I'm Corinne Moss. I'll be speaking about the effect of alcohol cleansing prior to vaccination. I have no disclosures to report. So Public Health Agency of Canada believes that you should be cleansed with alcohol prior to vaccination, while World Health Organization believes that soap and water is sufficient and there's not enough evidence indicating that you do need alcohol prior to vaccination. Through literature review, I haven't found much evidence to support either of these statements. So benefits of not swabbing is it reduces pain as alcohol can sting. It lessens fear as alcohol is a pretense to vaccines. Not using the materials is cheaper and it's overall better for the environment. Retributions of not swabbing is that it could introduce bacteria into the skin. So my study question is, what is the impact of alcohol application prior to vaccination on local skin reactions? So it was a randomized, double-blinded study. So it was with a group of people from ages of 0 to 18, and they were allocated into two groups, swabbed and non-swabbed. So the research assistant would enter the room, and he would rather swab the child at the point of injection or adjacent to the point of injection while blinding the parent and the child. Then the pediatrician would enter the room, and he would inject the child as a standardized procedure. Afterwards, the parent would be asked to have a study for 15 days post-vaccination, and a telephone interview was conducted day 1, day 5, and day 14, just to make sure there was no reactions or if the parent had any questions. So there was 170 participants around the age of 5.6. They rather had one injection or multiple injections, and there was no specific vaccine being used. And as you can see from the results here, there's no clear difference between the swab group and the non-swab group. And actually through all the non-swab group, there were no new reactions after the 4th day, and there's absolutely no cases of infections. So in conclusion, there is no evidence indicating that you do need alcohol cleansing prior to vaccination. And of course, this was a pilot study, so there are some limitations. Firstly, skin reactions were recorded by parents, so there could be misinterpretations of some symptoms. It's a small sample size, so it's hard to generalize the data to an overall population. And the timeline for data collection, which was 14 days, could be considered too short, even though in other practices, the 14 days to record cellulitis was sufficient. Parents recorded things such as redness, pain, pus, swelling, and other blinded pediatricians recorded things as abscess and cellulitis as per the Brighton collaboration criteria. So the next step to further this research would be a larger population size with more variety and demographics. Thank you for listening, and I hope this presentation led you to question the normal practice of alcohol cleansing.
Okay, thank you very much, and everybody stayed within the time limit, so this is great. We now have 15 minutes for discussion. If people want to ask questions, they can come up to the mic, and why don't I have all three of you come? We can't hear you very well, so maybe if you turn up the...
Okay. Thank you. All right, so this question is from Matthew online, from Melissa Perry. He says, excellent presentation. I was wondering whether you could give examples of misleading information about HPV that you found on PHU websites. Thank you.
Okay, great. Thank you so much for the question. A lot of the times what the misleading information was just outdated recommendations on who should get the vaccine, what gender, what age, and what vaccine should be utilized. Thank you.
I'll ask. Okay.
Oh,
go ahead.
This question is also for Melissa. Again, good presentation. You mentioned that the catch tool is made for written information. Did you look at written information that was sent home with consent for the grade 7 and 8s for HPV?
No, so it was originally made for paper products, but the reason for why we chose this particular tool was its comprehensiveness. So the catch tool in Nature has 12 distinct levels and 52 sub-levels, which range from topics including graphic layout and ranging from cultural understanding to scientific evidence. So what was done is each website was analyzed using that information, but we didn't get far enough to analyze the information that was sent home. But that's a great insight. Thank you.
Okay.
Okay, I have a question for Kate. So Kate, I think you showed us that about 33% of your of the total sample responded. Can you reflect on how that might have impacted the outcomes that you found?
Thanks for that question. So just first of all, the 33% response rate is actually very typical for those monthly mailings. That's sort of what they get for most of the surveys that they send out. But as far as in terms of whether that would overestimate, that was something that we were concerned about, whether people that had come across a child with a vaccine-preventable disease would be more likely to see this survey and answer it. But I think because we compared it to some of the national incidence estimates for the bigger population, that we sort of scaled down a little bit just to get a sense if it was in the range. It doesn't seem to be that would be such an overestimate. It's probably within the range of what we would imagine we would see.
Okay, there's got to be some more questions. Natasha.
So can I ask a question about the alcohol swabbing and what is happening? Have you got any sense of what's happening in other countries and jurisdictions? Or whether is this just a Canadian or an Ontario issue? Because anyway, I believe alcohol swabs have not been used in some of the countries for some time, so I just wondered if you'd looked at the international and national setting. Thanks.
So what kind of triggered this question is in 2015, I went to Tanzania and I kind of saw how their nursing stations were there, and I realized that they didn't have so many alcohol swabs. They weren't using it for all those patients, and that's what kind of led the cost issue. And I noticed in other in schools, when children are getting vaccinated, some of the nurses, the normal practice is to use 70% isopropyl alcohol, swab for 30 seconds, and let it dry for 30 seconds. But not all nurses did that. So if they're not using the procedure properly, then is it working effectively? If it's not working effectively, then do you actually need the alcohol? And that's what led me to the question.
So I might repeat that because I think there might be people online. So there was a comment on the Australian immunization recommendation that said, and correct me if I'm wrong, that as long as it's visibly clean, that there is no need to swab. Yes, but the evidence behind that can't be cited. Can I just get you to go to the mic, because it's really hard to hear up here because there's some background noise or something. Thank you.
Yeah, the question is for Corinne Moss, the alcohol swab thing. Your comparative sample, the comparative group, comparison group, why did you not do it with soap water like WHO recommends it? Soap and water?
Oh, so yeah, we didn't use soap and water because we just wanted to see how it would be strictly with no cleansing of the skin whatsoever. That was just our comparison.
Okay, and like even the adverse reactions in both the groups, the p-value was pretty insignificant, mainly due to the sample size. Did you consider before doing the study that you needed a larger sample size to do it?
So just to repeat, our sample size was 170 participants. This was a pilot study. Yes, that's a very small amount. We chose 170 participants because that amount of people would show the odds of showing a local skin reaction, not an infection, but a reaction. It was just a secondary outcome that there were no actual infections. To enlarge the study, to notice for an instance of infection, it would take 1 in 100,000 people, and to double that incidence of infection, so 2 in 100,000 people, that would take 4.7 million patients, and that would take around 27,000 clinics.
Thank you.
So a feasibility issue, maybe a little bit of a feasibility issue. Are there any more questions? Okay, well, if not, can you please join me in thanking the students for their wonderful presentation? Okay, now I know you need to put something. Yeah, but she wanted to put this up. And then we'll go to credit. Yes. Perfect. Okay, so for the next part of the afternoon session, we're going to be having 2 fabulous speakers with very different backgrounds, so I think it'll be great. And then a group discussion session based on the 2 speakers and a chance for you to reflect on the day. So we've got 3 reflection questions here that are that you can talk about more in the small group sessions. So, Lydia, now I'll get you to put up the next session. So our first speaker this afternoon now is Carly Weeks. And I suspect that we're all in for a treat. So Carly Weeks is an award-winning national health reporter and columnist at the Globe and Mail. She has established a reputation as a fearless journalist with a passion for shining a spotlight on the health and healthcare-related topics that matter to Canadians. We all have a vested interest in healthcare, and in a world in which we are increasingly surrounded by myths, misinformation, and lies that help to threaten to keep us misinformed and in the dark, Weeks specializes in finding the hidden stories that some organizations would rather not be told. She provides a healthy dose of reality in her writing and debunks myths on topics ranging from the prescription of painkiller crisis to fears over vaccination to big food and nutrition policy, to name just a few. She's also written about her personal experiences with extreme bullying as a child and has become a source of information and inspirations for families dealing with these serious issues. So please join me in welcoming Carly Weeks.
Thanks, everybody. And it's a real pleasure to be here, in part because I get to, hopefully converse and engage with you guys, and also because journalists rarely get out of the office these days. So, we're busy, busy people. And we're stuck to our phones all the time. So to see real faces is wonderful. So I'm just going to, which way's forward? One moment.
Is that going right? Yeah. I'm the worst person to do this. I have to. Do you have more than 2 slides?
No, I have 8.
8? It's being investigated.
Okay. I can begin. The slides are kind of tangential. So if they appear magically, we can move from there. And I can explain some of them if need be. So I'll just begin. So I've obviously been writing about vaccines now for a while, and I've been a health reporter at the Globe and Mail for about 12 and a half years, which is frightening how fast it's gone. And I've actually been around long enough that I remember a couple of years ago when an editor asked me to contact an anti-vaccine parent to get balance in a story that I was doing on a vaccine-related issue. I'm happy to report that that no longer happens, at least at our publication for the most part. But there are a number of issues and challenges that I think both public health and the media can address. And so I'm here today as a journalist. I'm not an expert. And what I would like to basically convey is some ways that I think we can all work together. So that question number 1 on the previous slide is sort of my wheelhouse today. And so, how we really can engage with each other to make for a more informed public. So we know that we're confronting a very serious problem. I could cite some very scary statistics that you guys are probably all aware of from Public Health Ontario. I recently was reporting on some of the new stuff that they have coming out, showing that in parts of Ontario, more than 10% of school-age children aren't properly vaccinated. Obviously, this is not good, and I think that there's a number of reasons why. So let's get into this. Should I wait? What? Okay. He's very close. I might wait just a second, because I just realized it kind of does help to have the visual. Okay, here we go. And yeah, this is basically, and then just that button, right? Okay. So obviously, this is what we're talking about today, when lies go viral. And this is the kind of thing that we all have to be careful and vigilant about. And I guess, what part of the problem we know is social media. We're going to talk a little bit about that. But also, this is an issue of framing, how we discuss this. And I think part of that is shaped by the media, and part of it is shaped by the information that we're getting. So again, no disclosures to make. This is often how we think when we think about vaccine-related stories, or at least this is how members of the public kind of see this issue. This is actually from an anti-vaccination website, and given everything that's gone on this week with the anti-vaccination movement using the photo of a dead child to promote their cause, I just want to be clear, I don't know who this child is or her backstory, but this is pulled from an anti-vaccine website, and this is the sort of imagery that we still see posted with anti-vaccine images on stories in the Globe and Mail and elsewhere around the world. And I think that this is something that needs to be countered and challenged. You know, that vaccines are not something to be fearful of or fearful about. It's something that we need to actually be celebrating. And so hopefully we can start seeing, I know, I'm not sure who would actually be having this sort of reaction during their vaccine, but yes. Yeah, so I think, you know, this was all that was available when I went to search for photos on like an iStock website. This is basically the only one that I could find that wasn't sort of really inflammatory with a giant needle. And so this is kind of what we're working with here, and I think we can agree that we can do better. You know, we need to be framing this as a discussion about, you know, the medical marvel that vaccines are, the things that they can do to help us. And I think that one of the reasons that those messages haven't come through as much as they need to is that journalists are getting the wrong messages. So this is actually a very old debate that just is coming up again. And so this is an article, the source is at the bottom, and so if you have a chance to give this a read, it's about victims of vaccination, and it's by, I think Catherine Arnup is her name, and she published this back in 1992. It was the history of the anti-vaccine movement, specifically in Ontario, and it's a great read, but I specifically highlighted this passage because, as you can see, there's an anti-vaccination league that posted a giant advertisement in a Toronto newspaper, and there was a backlash. And as you can see, the sort of medical officer of health of the time for the city basically said, you know, there's no point, these people are just ignoramuses. It kind of goes on from there, but like the recorded comments that he made at the time, that there's no need to even engage with these people because they're just too stupid for words. And this is self-evident that this works. And so, you know, I just wanted to leave that with you. Jumping forward a couple of decades, this is another story. This is from Maclean's, and I think this is from the mid-'80s, if I'm not mistaken. The citation's at the bottom if anyone does want it. So this is about a family, and it's presented as fact. This is a family who vaccinated their son. He appeared to have, and I think back then it was all about the whooping cough. Now, you know, it's MMR, then it was whooping cough. And so there was these scare stories coming out. We saw pertussis vaccinations drop, and obviously rates of whooping cough went up. But apparently this boy suffered irreparable damage. The family says that they will never vaccinate their child again, and they had another one on the way. She will never be vaccinated. I believe they had a girl. At the very sort of tail end of the article, they quote a sick kids expert who basically says vaccines are safe. But for the most part, this is how the media framed the issue for many, many years, continuing up until very recently, and even in some cases today. The previous article, I think, is an interesting illustration of how some public health officials have approached this issue, an approach that has in some cases still continued to this day. And again, I'm not here to criticize anyone, but I'm just sort of observing some of the things that I've noticed as a reporter reaching out to people on this issue. The interesting thing about this Maclean's article is that I kind of wondered what happened to this family. So I actually went on social media. I haven't reported on this, and I don't think I can, but I found this woman where she lives now, and I found her son, Steven, and he's like, seems perfectly normal, living his best life. He's getting married, like, you know, the things you can find out on Facebook today. I've reached out to them, and they wouldn't agree to talk to me. But I found that to be a very powerful example of how, in a lot of cases in the reporting that I've done on so-called miracle cures or, you know, these irreparable harms of vaccines are actually, they turn out not to be true. Now, that's not to belittle the stories of parents who have, you know, children who do have serious problems, whether it is autism or something else, or, you know, disabilities. Not saying that they're linked to vaccines, but I don't want to belittle those stories. But in this case, the media reports something as fact when it very clearly was not. When we think about sort of, you know, the previous article, I think that what I mean by saying that the public health messaging hasn't really changed much is that it has tended to be, you know, sort of the patriarchal, we know best sort of line coming from public health, from family doctors, from sort of public health units, that, you know, this is the information that you need. You don't need to know any more than this. The vaccines work, they're good, no questions asked, now you need to get one. And that's why there's always been sort of this push back against the idea of mandatory vaccination, you know, very fearful, what are you putting in me? This must be something nefarious if you're forcing everyone to get it. I think that the attempt was there to sort of shut down any debate or pretend that there was no debate to be had. But what sort of happened is that response, I mean, and by this I mean that I used to call people up and I would try and do stories on this, and it was hard to get people who wanted to really talk about the anti-vaccine movement. There's people that would talk about the effectiveness and safety of vaccines, or what the coverage rates are like, or, you know, there was a big debate, you know, that I wrote about a few months ago in Quebec about the number of doses of the HPV vaccine. But I think there was this real hesitation and fear to engage directly in the anti-vaccine discussion. Basically, are we just going to be amplifying them? And there's evidence that, you know, you talk about something and it does amplify it. It does attract people to it. It lets more people become aware of it. But I think what happened in the meantime, what we saw, is that at the same time, the social media was on the rise. And so what we end up with are these anti-vaccine groups and movements, many of whom have their own vested interests, who basically took that time to manipulate and figure out how best to use social media to attract attention. They basically became, you know, news entities in their own right? I mean, there's that Del Bigtree guy, I hate to even say his name, but you know, I interviewed him a few months ago when he was supposed to come to Toronto, and I interviewed him on the phone, and obviously he said I misquoted him and all of this stuff, and then he used that interview in a podcast that he had the next week. And so he basically played this recording of our interview, and he used the whole thing as fodder for his show. And it's brilliant. And he never wrote me a note saying, how dare you? I don't even think he was really upset by the coverage. I think it was just great publicity for him. And so, you know, that is obviously something, you know, that we have to be mindful of. But this is how sophisticated these individuals are. You know, and like I know from his tax filings, Mr. Bigtree makes at least $150,000 a year in his role with ICANN, his Informed Consent Action Network. They got over $1 million in donations last year. I don't know from where. We have places like the Children's Medical Safety Research Institute, which sounds like such an upstanding organization that is actually run by the Dwoskin family, who have had it out for vaccines for years. You know, these people are very savvy. They know what they're doing. They know how to get, you know, clicks and likes and retweets and all of those things. And sort of I've seen a lot of that firsthand. And it is, it's you know, scary because they're peddling, you know, half-truths, mistruths. And I think it was during conversations I've had with like Natasha and others in the room who basically said this is why they succeed. You know, there is a vaccine compensation system in the US. Vaccines aren't perfect. They don't work 100% of the time. And so they use this wiggle room to bring home this point, you know, that vaccines must then, therefore, be dangerous. And on the flip side, you know, we have our public health officials who can't engage with, you know, the same type of media manipulation. So it is a very difficult spot to be in, because on the one hand, you want the same number of people to be believing you as they do, you know, someone like Mr. Bigtree and Mr. Wakefield. But at the same time, you know, you have this responsibility to be truthful and accurate and all of that stuff. So I think that's one of the main issues that we have. Another issue, and this is not necessarily, I mean, this isn't related to social media, but something that we have seen that has certainly propelled, I think, the anti-vaccine movement, is that we do have a number of regulated health professionals in Ontario who promote these ideas. And I'm speaking specifically about some chiropractors, naturopaths, and homeopaths. So I did a story a couple of years ago, and I found that over half of the Toronto naturopaths, that was the only focus of my sort of investigation, were breaking their own college's rules by posting information online they weren't supposed to be posting, such as anti-vaccine information. And so when you sort of think about the credible sources that that's coming from, and by the way, every time we contact their regulatory bodies, they basically say, well, people can lodge a complaint, and the province says, yeah, well, it's up to the college to regulate. So it's this sort of really maddening circle that we are sort of running in. But, you know, this is sort of, you know, what we have been up against typically. You know, I think that if we do think that there's actually something good that's come from the recent outbreaks of measles, if there is anything good that's come from this, I think that it has sort of shaken everybody and woken everybody up, right from, from the media, public health, everybody, and we're realizing, okay, this is not playtime anymore. I mean, these people are a small minority, but they have a very loud voice. And once you're exposed to these kinds of ideas, it can be very easy for these grains of doubt to sort of seep in, You're at the doctor's office with your child and you're thinking, wait a second, are these things safe? You can see how easy it is for the public to start to lose trust. And of course, we're in an age where, mistrust of official authority figures is at an all-time high. And this is something that we see can be even more true in certain segments of the population that may be harder to reach. Newcomers to Canada, for one. And we know that there are certain, religious communities and others that have some of these same things at play. And so that's why, there's an outbreak or there's 2 cases of measles in New Brunswick right now, and one of them occurred at a student was at a school. He or she contracted measles at an emergency room, but then was at school and at a big John Cleese show. And so now, is the other shoe going to drop? We kind of don't know. And so, that's why obviously there are so many concerns. And this slide I wanted to include because what I've seen, and in light of these outbreaks, the good thing that's come is that public health professionals, media, others have really upped their game now. My editors are happy to put anti-vaccine stories on the front page to counter these myths. And at the same time, now when I reach out, I immediately can find experts who will sort of directly engage and counter the lies that this movement is spewing, but also to repeat the positive messages about why vaccines are so great and why they work. So this particular story, this is a wild one because I was following with the Vaccine Choice website for a while on some other stories that I had done, and I looked at their Facebook page on a Saturday night, which I guess tells you something about the state of my social life right now. I have a 2-year-old, so yeah, we don't get out much, and I have a bit of a cold too from him, so sorry about that. But I was checking out their Facebook page, and they were like congratulating themselves on this billboard campaign, and I thought that it had to be made up. Like there was no way that they really had billboards up, and then all of their members started to post the billboards. Oh, I just came across one here, and I came across one there, and someone finally said, I found this one at, like by the Eaton Center, Yonge and Dundas. So I actually went there. Like I just bolted from the office and I stood there until I saw the ads with my own eyes. And only then did I pitch it to my editors because I thought there's no way this is real. And of course it was. The reaction was swift. I had Public Health Ontario, Toronto Public Health on the phone in minutes, some amazing sort of quotes, and even the health minister ended up weighing in the next day. I mean, and I think that this is an example of how these things can work, how we can work together to counter a lot of these myths and to send a really powerful message across. Because I was sort of looking at it as a bit of an analogy, so if social media is a theater, the anti-vaxxers have had, center stage for some time now. And now it's up to the rest of us to sort of play catch up and figure out the best ways to do that. The Public Health Agency of Canada has recently said that they're going to be starting a counter campaign. They're working with social scientists to sort of battle some of these lies and figure out effective strategies to bring people around and believe, and not just read the information, but actually believe what they're seeing. This is one recent slide that the CDC posted, and obviously this is something that they're attempting to do. You're basically using that emotion to get a response from people, right? You're not just saying vaccines are highly effective in 98% of cases or whatever it is, or only 1 in a million people might have an adverse event. This is something that's saying this is what measles looks like for everyone who forgets, Now, the reason I wanted to highlight this is that you might notice some of the comments on the side. They're a little bit hard to read, but they are all from anti-vaccination believers saying, this like CDC's lying, this isn't even real measles, this is just fabricated, there's no new cases of measles to even be reported, like it's all made up. And all of those comments, there's hundreds of them, and none of them were answered by anyone from the CDC. They're just up there, and it's the first thing you see when you go to that post, which is fairly prominent on their page. So I guess the lesson here is that there's more that we can do, you know, as media, as public health, others, to sort of be these guardians of truth and people who are spreading, you know, good information, not bad. You know, I guess in a nutshell, social media is no longer the intern's job. You know, this is something that needs like high-level priority. This is where people are. This is the 21st century, and so this is how we have to engage with them. You know, they use compelling horror stories of tragedy that tug on heartstrings, and they used to generate front pages. Now they generate thousands and thousands of pages, likes and clicks and shares. You know, it doesn't matter necessarily what the Globe and Mail says if, you know, 50,000 people who are on, you know, Robert F. Kennedy's site or one of the other anti-vaccine sites, you know, sees this information and believes it. So we need to fight sort of this battle on that playing field. And so what does that look like? That could be finding people that have been left paralyzed by polio, you know, showing people that have suffered as a result of vaccine preventable diseases and illnesses. What does it really look like? Let's trigger that emotional response in people. And that's not to say this needs to dumb anything down or, you know, sink to anyone's level, but this is the arena where people are living right now. This is where people get their news and information. Sadly, it's not necessarily from credible news sources anymore. And so, you know, images such as these can go viral, but then it also has to be, you know, the follow-up and the response to it as well. I think, you know, for too long we saw messages from, let's just say, you know, the CDC or Health Canada that would basically just remind us to get your vaccine. Vaccines work. You know, and people, we know just that information just kind of goes in and out. It has no real impact. So I would argue that, you know, really compelling storytelling is part of this. And that's what we do as journalists every day. You know, people often wonder what makes it into the newspaper, and that's it. Something that is compelling, new, interesting, you know, something that tells us something we didn't already know. You know, that you guys are probably all familiar with the example of Jill Promoli, who she's become a real social media presence. So her very sad story, one of her sons died after getting the flu as a 2-year-old, terrible. So and he had been vaccinated, but her other child brought the flu into the house, infected all 3 of her kids. He didn't mount the immune response required. He got sick, he died. Very sad. But she's used, if you want to check her out, she's at For Jude for Everyone. And she's basically become this national voice for vaccinations. And I wrote a story a few weeks ago. She's been under attack by them many, many times. Most recently, her posts were shared on an anti-vaccine website, and she had, you know, hundreds and hundreds of people attacking her, saying that she was lying about the way her child died, and I mean, just terrible things. But she says that, you know, the reason why she does this is because she really believes, you know, and she's never going to stop. And so those sorts of things are commendable. And so how can we all kind of, you know, leverage these stories, find new ones, and really sort of get those messages through to people? I know that there's a lot of work that needs to be done on the ground as well, you know, actually reaching out and physically contacting people that might have some of these doubts and things like that. But I think that using some of these counter messages is a very effective tool as well. And I certainly know every time I think about the flu shot, I think about Jill and her family. And I think that, you know, it's an example like that that can really go a long way. I think what we're trying to do is reach those people in the middle, you know, not the hardcore people that might never change their minds, not the people that are already convinced and know better, but those lot of people who are in the middle that are kind of just watching and seeing. I think it was an Angus Reid survey recently that said 1/4 of Canadians believe that vaccines are risky, that they're not safe. So there is this element of people that are on the fence, and you know, this is what needs to be done in order to hopefully close that gap. So with that, I will draw everything to a close. Thank you for listening and for dealing with the technical difficulties at the beginning. So, and I think we'll have a discussion later. So thank you for that, and please let me know if you have any questions.
We have 10 minutes for questions now. Is there a way to get the microphone so that Carly can sit here rather than stand here, or no? You can stand? Okay. Does anyone have any questions? I know we have at least 2 people in the room that have been subject to kind of vicious anti-immunization. attack. So, oh, Vanita.
Oh, yeah. Okay, so my name's Vanita Dubé. Carly, I wanted to introduce myself with you because there was one point when I was talking to Carly almost every day, and I felt like I was her best friend.
It was wonderful.
She was my best friend, actually, because she was putting the stores out. A couple of things. Can you go back to the slide with the billboard ads?
This one?
Yeah, so look, it's interesting because if you look beside the ad, it said, "This is off the wall." I've seen that so many times, so that's the first time I've noticed that. So anyways, I think this reporting that you did on the billboard ads was, I mean, you were the one that ended up causing the billboard ads to come down. And so I commend you for that, because this was truly all your hard work.
Oh, thank you. That makes my day. Thank you.
Yeah, because had you not reported on it and then it spiraled, so anyways, that's really great. Thank you also for bringing up For Jude for Everyone. I just wanted to share an anecdote that I had actually used that website and some of her information for some training that we did for childcare operators. And there was one point where Toronto Public Health was recommending that childcare staff get the flu vaccine. And in the comments from these childcare operators, it said, you know, the information about the flu vaccine causing death in a 2-year-old was really out there. And these are not necessarily, I mean, it's interesting because some people have particular opinions about the flu vaccine, but it's kind of, yeah, like sometimes it's great to go strong with a message of this is what vaccines prevent and guess, and if you don't get it, these are what the diseases look like. They can, you know, end in death. And on the other hand, then you get the other side, we're saying you're being really, really ridiculous. You know, be a bit more like, is it really that bad? Or you know, you're hyperinflating things. So finding that balance.
Right. That's a good point, and I know it's something that's come up with the measles discussion. You know, there were 2 cases in Toronto this week, I think it was, and you know, we didn't sort of I didn't write a standalone story. I think we just let sort of Canadian press cover it. You know, I don't think we want to, sensationalize everything and basically say this is, oh my God, we're all going to get measles, but I think that, you know, the media still often has to find its way, and it's not like we're all speaking with one voice. Everybody has. I mean, the Toronto Sun published an article by like that wacko doctor a few months ago promoting crazy anti-vaccine views and his own vitamin company. Like it's 2019 and that still happens. You know, but yeah, like it is a good point because, you know, you don't want to scare people into sort of like, you know, shock and, you know, they kind of turn off the alarm, right? Like the alarm fatigue. But at the same time, I think some of those messages, certainly I think from our perspective, like mine in the media, you know, we haven't done enough in that arena, you know, that there does need to be more to kind of, you know, educate people and use some of those, you know, stories or whatever, you know, without bordering on the sensational. It's a constant balancing act that we have to juggle with, but I'm just grateful that, you know, we are at least trying that now. You know, I think it was 2015 that Toronto Star published the Gardasil article, which, you know, it still boggles my mind, you know, that happened, but it did, and so, you know, I guess the other thing I would just say is that, you know, we're only as good as the information that we get. And so I thank people like Vanita, like Natasha for taking my calls and making the time. I think it was Michael Gardam a couple of years ago who was telling me, I was speaking to a public health group of students and I was asking him, you know, what is it about, like why do you make the media a priority? You know, because a lot of times people don't want to take our calls, just one more thing in my day, or they're going to misquote me, or it's a fear. And he said, you know, if I'm in a meeting or whatever about SARS or whatever it was, you know, he's recounting what in the past, he realized that taking that call from the Toronto media is probably the best thing that he can do all day to get the right and correct messages out, right? So without that, we're filling in the blanks from other sources, right? So that's, so my constant message is just, you know, please take our calls.
So thank you very much for the presentation. I actually had a slight comment / question about a lessons learned that maybe we could take from you. And it's about, I mean, you're standing in front of a room of scientists who are very good at talking about data. But what you mentioned is about how we tell stories and how compelling that becomes. And I'm just wondering if you could share your thoughts on how we could leverage some of that storytelling ability,
right.
Whether it's at our at our basic education level, where we're starting to have to train not just to be able to debate data amongst ourselves in a traditional scientific format, but recognizing that that data doesn't sit inside those closed rooms anymore, that it's easily accessible outside. And should we actually be changing the way we educate our next generation of scientists? You saw some very good presentations just recently. What are the types of lessons that you would want to get them to understand about how do they make their data more accessible, yet and compelling, I guess?
That's a very good question, and it's very relevant. Storytelling is powerful, but it's a skill set, right? I mean, and it's something like, this, what is a news story, right? What makes it onto the front page? It's sort of, it can be in a bit of an elusive formula. But I think that in a dream world, let's just say that there's, all of these resources and things like that, you might have someone who is on a team who's dedicated to helping you sort of figure out how you can pull some of the people out of the information. Now, obviously that's not possible in a lot of cases, and there's always budget constraints and things like that. So I think a couple of takeaway points, and hopefully this is helpful. As you say, data can only just sort of point to a story, but there are always people behind that story. So maybe it is sort of looking into the past. What is the data? So if we're seeing data, and we are in Ontario, that there are certain communities that aren't being vaccinated, likely due to fears that have maybe been stoked by some anti-vaccine people. I mean, we know that the New York measles outbreak, a lot of the communities were actually targeted by anti-vaccine groups. I don't know if that's happened here at all, but let's just say, you can maybe create a campaign or some sort of targeted messaging for some of those groups using some examples. I mean, I find that we always talk in journalism about the real people. Like, oh, do you have a real person? And it might sound so strange, but it's like you're telling a story about some new study that just came out about how some drug or some new way of doing things is going to help people's lives. But unless you have a real person, that story is going nowhere. And so we spend a lot of our time actually looking for these elusive real people. And so that's something, at the very basic point, like whether it is, just something about vaccines or something else, figuring out who could be a person to tell that. Is it a parent, someone like a Jill Promoli, or is it someone who suffered long ago, or someone in between? Like maybe it's someone who's doing something really innovative and who has a really compelling story to share, So I think that, oftentimes we get, say, pitches from people, from PR companies or from hospitals or things like that, and then a lot of them end up not being very great, I think it's because, again, a lot of people don't understand what is news, how do you make a news story, and so what I always tell people who ask for advice is that you want to really flip the equation on its head. You should be spending like about 90% of your time on figuring out what is the story, how are you going to tell it. If your goal is public engagement, 90% of the resources should be dedicated to figuring out what the most compelling story is and how to tell it. And the other 10% is sort of the execution. Now, that you might need to tweak those numbers a bit, but the point is that the figuring out what the story is more important than I think. I can't overstate the importance, because without that, if you can't tell me in one line why I need to care, then no one else will care, right? And can a 12-year-old understand it? That's the other takeaway that we often have. Hi.
Hi, how do you do? Lawrence Lowe, Associate Medical Officer for Health at Peel Public Health. So really great to hear that, at least that piece around the newsworthiness of stories. One thing that I've also noticed, at least in terms of newsworthiness of stories, is that it tends to be mostly trending towards, the negative, right? Like these are, vaccination rates are low. The Gardasil story that you mentioned in the Toronto Star is a sentinel example. And I guess the question I have is really one of the great good news stories out of vaccination, and also the challenge that public health has as a whole, is that when we do our job right and when things go right, we're almost always invisible in success. And that's the good news story. So I guess the question I have is, with that good news story or other good news stories that we might have in vaccination, how do we make those newsworthy? Yeah. Because nobody really cares about the invisible sometimes.
That's right. That's a much harder question. So, thanks for coming. But it is. It's, oh, the sky is still blue, the grass is still green, everything is the same. Like, it's true. Like, it used to be, if it bleeds, it leads. And we don't really live in that world anymore, but we do. We gravitate toward the controversial, the unexpected, the negative, the news, right? Vaccination rates are low, people could get measles. There's a couple of different ways, and this requires a lot of dedication and resources, and it's again, it does go back to that formula of storytelling, but I think in that kind of a view, if there's a certain project or milestone or whatever it is to highlight, don't just highlight the milestone. I would say again, go back and figure out, how can we tell this story? Has this really led to some sort of change? Is there someone who maybe works in our office for, like 50 years who's actually can remember back in the day when we were doing some really ancient technique, and we can actually bring you in and show you some old, like out of date thing that we used to use, and now this new technology. Or again, going back, people are always very powerful. Examples of people that have been helped. Someone who actually, like, their life was made better because of this. Oftentimes if you have a personal story, like a 9-year-old so-and-so who, was managed to improve her health dramatically, or whatever it might be, whatever the story is. We're sticking to the example of vaccines. Like I will always sort of say I do think that the people aspect is most important. Saying that, you have a 2% uptick in vaccination rates among 7-year-olds in this district, probably not going to be generating headlines, but, you can flip it on another way. You could say, you find someone who's coming in, and again, it's difficult, it's hard, but you find someone, maybe you know of someone who knows of someone whose child wasn't vaccinated, now they are. Those kinds of things. And in the business like that I'm in, we spend a lot of time doing that, and it can be very hard, but in the age of social media, it's not so hard. Go to Facebook, go to community groups, find some of those people in your community who are actually changing their minds, or whatever it is, those kinds of things. Leverage the people, leverage the tools to then come to the media with those stories. I hope that helps. And you can always feel free to reach out to me on Twitter or anything. I'm available. Yep.
I'm just aware there's a lineup behind me, so I'm going to be really quick. You asked why people are reluctant to come forward, and I just thought I might just comment on that. I mean, one thing is it's actually very scary for most people working in public health. I always get absolutely terrified about going on the radio. It's one of those things that I find really hard and stressful, but feel I have to do. So, and partly that's the UK history, because the press in the UK, I kind of grew up under the MMR and autism scare, and the press were awful there. So, but I was on the radio, I've got this list in front of me. I was on the radio this week. I won't say who was interviewing me. And well, okay. So I've had really good experience, including with yourself. And I wanted to say that the journalist you're working with is really important. I got kind of ambushed on the radio by somebody from outside this province who started off by saying she was pro-vaccine, and then went through this list of things, and without prior agreement had questions coming in from callers about why we don't have a compensation scheme, why we're not allowed to vaccinate children under 24 months. These are bizarre questions. What's the point of vaccinating if vaccines wear off? Why bother with a flu shot when it's not effective? Isn't there a problem if you've got a family history of autism? You may still get autism from MMR. Oh my God. And a vaccine. And why do we not talk about the fact that vaccines are not 100% safe? And so those are all individually not unreasonable questions in some ways. I mean, some of them are just daft, but they were like fired at me like a sequence of, we've got one caller who's asking this, and and so just to say that I think we need good journalists like you, who we have confidence in, who's going to be a good journalist. And I'm not saying you're going to believe everything I say. I'm happy to be questioned. It's more that we need to be able to trust the journalist as well, because you can't always do that. So.
And people have been burned in the past, and you're fantastic. I would have no idea that you're at all nervous, because you're a fantastic interview, and it just shows that sometimes you do need to actually. It's a skill on both sides, right? There's no sort of journalism like accreditation. I mean, you get hired into a newsroom, you start working there. Some places, and I'm sorry for that experience, but there are people that will. There are negative experiences to be had. I don't want to single out any person or entity, but of course. The only thing I could say to counter that, and again, it's not a foolproof solution, but like my brother actually trains people in media. Like he was sort of a journalist early in his career, now he's on the other side. He trains people before interviews, and we talk about this stuff all the time, and it just infuriates him when he sees the attack. I'm like being attacked by like the anti-vaccine people and the pro-gun people all at the same time. So it's like exhausting these days. Oh my goodness. But it's, you know, this idea of being prepared sort of for anything, you know, and of course hindsight being 20/20 and all of that. You're finding, you know, sometimes there are going to be bad apples. You're being interviewed by someone who has a clear axe to grind, who has a clear agenda. And the only thing that, I mean, and I've been interviewed myself before, so I've been in those shoes, and the only thing that I try to always remember is that, you know, this person might not end up being the person that they say they're going to be. They might, you know, and I think it's the only way. Like, there will be bad experiences, but hopefully more will be good.
Okay, I'm sorry, but we're over time. And so I'm getting the signal here. So what we'll do is there's another discussion period at the end. So if I can get you guys to sit down, I'm really sorry about that. But would like, no, you don't need to apologize. I'd like to take another opportunity to thank Carly for coming. I think that was a fascinating talk.
Thanks very much. I'll just stay up here for now.
Yeah. So Carly indicated she'd stay around so that if there are some questions that we can, she'll take those afterwards. Okay, now we're also in for a treat because we have Dr. Stephen Moss. Stephen is a pediatrician who on the basis of his work in the area of education and immunization promotion was promoted nationally to associate professor at the University of Toronto. His clinical work at the Hospital for Sick Children and in the community. He served as chair of education for Child Health Network, pediatric education director at North York General, Foundations of Medical Practice lecturer for University of Toronto Medical School, pediatrics chapter editor for the University of Toronto Notes Text, and sat on Ontario's Provincial Infectious Disease Advisory Committee on Immunization between 2011 and 2014. Yay! He his recent randomized control research publications have been in the area of pain reduction in vaccination. Stephen has earned many teaching awards in varied areas of academic education, and that's not all. His volunteer work has taken him to the interior rivers of Guyana, an orphanage for HIV-infected children in central Tanzania, and in 2018, I'm going to pronounce this wrong, Aboizo, in West Africa. So please join me in welcoming Dr. Moss.
Jenner, on the upper left-hand side of the screen, has been cited as the single human who has prevented the most illness in the history of the world. Yet, he also had people that didn't believe in his practice either of immunization. And later, I'll invite you to come up and read some of the comments here. They're still being echoed today. The truth is that if you asked someone who was vaccine hesitant, and you asked them, just give me the summary. Like, what's the summary? What's the big picture? Carly actually just told us the big picture. It was the feedback that was done on the internet. Basically, the CDC is lying. And basically, it's all made up. That's their summary of how they feel. And today, we're going to address those issues and talk about how we as providers could actually be part of the barrier towards vaccination in and of itself. I want to start off by asking you to answer the question, because almost everybody in this room is a healthcare provider of some sort. You're all involved in healthcare provision, and you're all knowledgeable about immunization and vaccination. So in your mind, I'd like you yourself to answer the question, and then I'll show you the answer to a survey of family doctors who were asked about their most significant factor in not recommending HPV. What they did is they looked for family doctors who had low rates of HPV recommendations, and they wanted to know what the factor was so that we could develop a program on how to answer them. So in your mind, think to yourself, is it 1, is it 2, is it 3, is it 4? So the only answer that came out was the gender of the doctor. Thank you. So you've just answered yourself your own question, that even in our own minds, of people in our own room, we are unclear of what providers are really thinking. What the study represented was that the female family doctors recommended HPV, but the family doctors only had a solid recommendation for HPV immunization in only 55% of in their visits. Whereas the female family doctors had 100% recommendation for HPV for women. So the difference was not about knowledge, and it wasn't about recommendations. It was inherent to the provider themselves. So with that introduction, I don't have any specific disclosures for this talk, but I have received honoraria in the past from Merck and Pfizer. And the objective today is to fulfill the fourth objective of the conference, and that's to talk about how we can improve communication about vaccine hesitancy. But in this framework, it's not going to be the public. It's going to be the providers, the people in this room themselves, including me. And the reason why I feel close to this is because we know statistically, we know the data, MMR, you give it, you get rid of measles. The graph on your left demonstrates that. Yet, I have a patient in my practice whose mom was not vaccinated because she was part of a family that didn't vaccinate, and they had this baby in Toronto who got rubella. And this is a picture of Artie on your left with permission, who I had to hospitalize at North York General and put a G-tube in. And this is an example of the dichotomy of issues that we're going to discuss today. Let's answer the next question. In another study, they asked healthcare providers, what route do you think the patients and caregivers would most likely want, if they had choice, to vaccinate their child with? Would they choose a needle, would they choose oral route, or would they choose nasal route? This is asking healthcare providers what they think their patients would like. So in your own minds, think about which would be number 1, 2, and 3. Timer's up. Pardon me? So this was for pediatrics, and it would be based on children. But they asked family physicians and pediatricians. Yes, it would be children up to the age of 18. So they were asking in general. Nobody cares about adults anyways. So if you were asking family doctors and you're asking yourself, which route do you think that patients between the ages of 0 and 18 would most likely prefer to have their immunization route, should they be able to choose? There was a resounding answer, and the answer was needle. Again, listen to the room. Wow, we're all healthcare providers, but we are not sure what our patients want. When this study was done, and in the commentary, they wrote down some of the thoughts. And one parent said, I just want it done. I want to see the needle going into the arm and know that my kid's immunized. Enough with this liquid dripping down the chin. Enough with this nasal spray getting mostly on my eyes and not in my kid's nose. They want the needle. In other words, how the vaccine is given is not the crux. It's not the real issue. It's part of the issue. And yes, how the needle's given, reducing pain, all very important, and we want to focus on that, but it doesn't answer what an anti-vaccinator thinks in their mind. If they want it not done, they don't want it done. If they do want it done, they want it done. Give that needle. We are unclear as healthcare professionals what our patients want. Now, how badly are we doing at this? Let's say a patient doesn't want to get immunized in their practice. Are healthcare providers in the community able to cope with this? Are pediatricians able to cope with this? No, we're not. We make a mess of it. And this is what we're battling against. Let's say that you wanted to figure out, okay, we're healthcare professionals, we can get around this. Let's figure some practical things out. And let's say you did a chart review. I love these studies. Chart reviews are the best. You go into somebody's office and you review their charts. You don't talk to them. Nobody cares what they have to think. You just want to see, did you give a shot? Did you not give a shot? And they found certain groups of practitioners that were very successful in immunization, like up to 100%, and they found some that were down near 40, 50%. And they wanted to say, give me a practical thing that I could give advice to help correct this. The single most important factor was giving multiple injections in a vaccine. There are many pediatricians in Toronto who do not give 3 shots at the 12-month visit. They move one of the vaccines over to the 15-month visit because they feel the healthcare practitioners feel there would be better acceptance. Maybe we can name names later. So the truth is that if you did a chart review, you would find that they would be less successful in immunizing their practice than they would if they followed a protocol. The varicella vaccine was the brunt of all vaccines lately. I remember when I would give discussions to people about vaccines, we had Menjigate, we had Prevnar, we had Varivax that had come out. All 3 of them came out at once. Menjigate took me 3 minutes. Yep, everybody's going to get it. Prevnar, pneumococcal vaccine, got to give that one. Varivax was about a half-hour discussion. But what about this? Is it better to get the immunity from a chickenpox party? How do we know that it works? Question after question. So we wanted to look at why there was this issue in the community. And one of the things that came up was actually that they didn't want to give so many vaccines at the same visit. They were worried about multiple doses of vaccines. Even though the data is quite clear that we're actually giving less antigenic load now than I got when I was a baby, that data is not that important. It's the feeling of how many needles. And changing that one practical aspect could make a big difference. Now, we're healthcare providers, and I have a very simple statement that we know that the greatest reason for impacting a patient's disease manifestations and how they manage it themselves is a healthcare provider's recommendation. And in the studies, healthcare provider's recommendation is the single most important factor to a person who's receiving a vaccine, like a caregiver or a patient, about their decision to make the vaccine. We can all agree on that? Sure, we're all healthcare providers. Good. Yet, how do we change practice? Do we educate people? I've heard some calls in the room about education. Yet, Carly told us that, I don't know about education because the truth is you're all lying. CDC is lying and it's all made up. So does education make a difference? I was part of the Child Health Network. I spent literally 10 years. We had 20 nurse practitioners and working on an education program to see if we could change surgeons from giving intramuscular Demerol to IV. And I will tell you, at the end of that long haul, it made no difference. We could not educate them. These are surgeons, of course. We could not change their minds. It basically took the pharmacist to change the order from Demerol to morphine to affect any change. Now, let's say I wasn't so smart, nor was my group, 10 years of my life, don't worry about it. So we took another approach, and we looked at vaccine education in general. Does educating healthcare providers about immunization improve vaccine uptake in the practice? So in this study, they looked at 56 clinics, 340 mothers. Their interventional program, nice, fancy, ask, acknowledge, advise. They gave food here, so people would listen. And they had 45 minutes of didactic stuff. They had interactive sessions. They had video. There was a pediatrician, vaccine expert, and a public health nurse who gave these talks to these physicians. They got materials, they got buttons, they got mugs, they got a 3-month follow-up, and they got monthly emails. The control was nothing. You might have well guessed it made no difference. There are many studies like this. So now we're thinking to ourselves, oh my God, what are we going to do? Because education doesn't seem to make a difference. So providing all this education and all this data about how vaccines work and how they don't have side effects, for me, it doesn't seem to make a difference. It certainly doesn't make a scientific difference if you reviewed education as a factor in changing outcomes. What do you think healthcare providers could be thinking to themselves? When you look at this, providers, when you think of asking the questions about vaccine hesitancy, the providers say it's the patient's concern about side effects. It's their concern about needles. It's their concern about cost. Yet when you talk to the patients using the same data, they report that their healthcare provider doesn't make good recommendations. And they mistake what the needs are of the patient and what the concerns are of the patient. So again, the scientific literature shows a dichotomy between what the healthcare providers are thinking and what patients' needs are. Another study looked at healthcare providers who were providing vaccines to their patients, and they also looked smartly at healthcare providers receiving their own vaccination and what their beliefs are. And they wanted to look at the groups, of course, who were less recommending vaccines to their patients and the groups that were less taking vaccines themselves. And they wanted to figure out what were they thinking. And when you look at them and you looked at the healthcare providers who were not necessarily vaccinating all of their patients compared to the other group, aside from whining about lack of time, the most major factor was the healthcare providers' fear of adverse side effects and vaccine safety. This is what the healthcare providers felt. When the healthcare providers may not have taken the vaccine. So we all have this issue about whether healthcare providers take influenza. It was brought up today. If you asked about all of the reasons why wouldn't you take the influenza vaccine, the number one reason shown on your left is fear of adverse effects and vaccine safety. It's the healthcare providers that are concerned about side effects. It's the healthcare providers that are concerned about safety. They have their own concerns. So what can we do about these guys? Well, and gals. They were giving the HPV vaccine. So what can we do about these people? Can I use people? People of all genders and types, or non-genders, who view vaccines as so back to this, healthcare providers, who view vaccines as risky, lead to reduced recommendations for vaccines. So that's still number one in this study that looked at what are we going to do with these people. Healthcare providers who selectively oppose combinations of vaccines like MMRV, individual vaccines, the pertussis, the MMRs, the usual suspects when it comes to these talks, and HPV vaccines, which healthcare providers in this study called lifestyle vaccines. These are healthcare providers' words. Lead to reduced uptake. Personal things like, did you get vaccinated yourself? We know this is a very common thing that people ask. And there was a study looking at midwives who they vaccinated themselves, they were likely to recommend it. They didn't vaccinate themselves, they didn't tend to recommend it. So this is, now we're getting to a list of things that we can really start addressing. Their recommendations in general were altered by their perceived susceptibility of getting a disease and the severity of the disease, their immunology beliefs, norms, industry, and personal experience. I'll come back to those. Their personal beliefs as well, like do I need to discuss sexuality before giving the HPV vaccine? These are not thoughts. These are the outcomes of different studies in the literature, which are all cited below, about what they're actually thinking when they're not quite recommending vaccines to their practice. So as we conclude here, what can we do as leaders? In one study, they looked at British, European, American, and the Canadian literature, and they reviewed all of this data, and they wanted to see, well, is there something that we could do as leadership? Their answer was to build trust, not information and education. And it seems to me that most of our efforts are around providing information. Good information, we say, the proper information. But remember, the other side is just saying, you're lying. Show me your data. You're just lying, you made all that up. Measles didn't get rid of, the measles vaccine didn't get rid of measles, sanitation got rid of measles. The most significant factors were trust of the information source rather than the access to the lack of information or misinformation. So it wasn't that people felt that they didn't have information, they didn't trust it. Trust of the claim and the legitimacy of the policy. So this went even deeper. The CDC lies. I just learned that from Carly today, but it's great for my talk. Okay, that's this comment. Legitimacy of policy. We need to help people understand that policy is legitimate and that the claims are truthful. People don't believe it. There was another interesting thing that came up, which was new to me, this acknowledgement of individual empowerment and choice. New to me because I didn't grow up this way. But now there's an empowerment of individuals and choice of individuals that the medical community and the rest of the world is looking at. You could have a choice. You're the one in control of your healthcare. You could go to Life Labs and look up your own results and make your own conclusions about what your lab stuff shows. Sunnybrook does, all the hospitals, many hospitals do, and they're empowering patients to take care of their own health care. We have to jump on that bandwagon. So rather than giving out a lot of data, maybe we should go back to what Jenner was commonly known to do, and he would sit with the farmers talking about cowpox and smallpox and engaging them to join him in trusting him that he may be able to help their milkmaids and not acquire disease. So in discussion, I hope that we can improve vaccine uptake by adding a focus on healthcare providers themselves. In general, I hope to build trust of claims and legitimacy of recommendations, engage this movement of empowerment and individualized healthcare, broaden the inclusion. We might think about speaking not just about patients who are vaccine hesitant, but healthcare providers who are vaccine hesitant and alternative healthcare providers. Maybe we could think about complementary and alternative medicine joining us in a research trial of side effects of vaccines, where they could be the lead author and we could be assisting with the research in some way. Let them become leaders in engagement of producing their own data, basically, that they may believe themselves. Specific things are translational. Translational meaning translational education. Talking about side effects, severity of disease, effectiveness, addressing beliefs, addressing the concept of, is it better to get measles and be protected, or is it better to get vaccinated and get protected? Another crux of the complementary and alternative medicine argument, as we know. What is normal? Patients in my practice who don't want to take vaccines, they think they're normal. They think, oh, there's lots of us that don't want to take vaccines. We're part of the norm type of thing. Industry, skepticism of industry, skepticism of government, personal experiences with the vaccines, and of course, all the practical things that we think that we can do, like basically even just adhering to an immunization schedule to make sure that we don't miss people who are vaccinated. I hope that in our future, we could take some of these articles, like the McLean's article, and fuss it around a little bit, add a couple little things to make it look like this. Our girls are at risk of cancer, and inoculation of Canadian girls against a sexually transmitted virus is underway. We could stop cervical cancer. Thank you very much.
Okay, thanks, Stephen. And we have some time for questions.
Shelley and I will be happy to answer any questions that you have.
Yeah. Thank you. Thanks, Steve. Actually, your presentation really was just summarizing what I was going to say to Carly's great presentation in some ways. And it's around the messaging. And I think this is where the sociologists and the psychologists are really valuable, but you've shown it with some really hard evidence, is that humans are driven by really two things: fear and trust. And when you can combat the fear, or address the fear, or use the fear, but then have a layer of trust on it, doesn't matter. HCPs are human beings as well, and we all biologically potentially work the same. And I think the other thing around the messaging is, and a really good example is what Public Health England is going through at the moment in terms of explaining their meningococcal campaign in infants. So they have some vaccine effectiveness information or results, which you would go, okay, they're good, but maybe not outstanding or as much as people expect. But what they've done is they've actually looked at that result and said, well, this vaccine effectiveness really translates to one child being prevented from having meningococcal disease every 4 days. And that actually resonates much more strongly than saying, well, the vaccine effectiveness is 60%, 70% with confidence intervals this broad, blah, blah, blah. No one gets that. But when you say a baby's protected every 4 days, everyone knows.
Nice, thank you.
Nice presentation, thank you. I just wanted to follow up on the fear and trust issue, and we have pediatricians, I know of several of them in the US, that are working, they use people's trust in their education and their knowledge level, and yet these pediatricians are spreading fear, being anti-vaxxers. What do we need to do to work with those people so that they aren't out there increasing fear about vaccines?
Well, I hope the discussion period will elucidate some things from the audience. From my standpoint, I think we should be engaging them rather than disengaging them. I think we should be inviting them into our world. And I think we should be having discussions, hearing what they have to say, and helping them themselves figure out whether there is some truth to it from their own perspective. I would love to see us helping them prove that vaccines are safe or unsafe. Let's let them do that study. Let's let them follow people moving forward. Let's let them look at vaccine efficacy. Let's get Spears in here and say, you know, you were once allowed to practice as a physician, so now let's bring that back. And I think that engaging them might be the solution.
We can get you to go back, and then Elizabeth, and then we'll go.
Hi there. Thanks, that was great. Actually, they were both great. I have another specific thing for your kind of what do healthcare workers think about vaccines. One of the things that we find all the time when we're doing contact follow-up when there was a vaccine preventable disease exposure in a healthcare setting, people have no idea whether they've been vaccinated or not. They have no idea where their vaccine records are. Physicians, nurses, office staff, you name it, they have no idea if they've been vaccinated or not.
Absolutely.
So it's even more basic than what do they think. It's like, do they even think about it at all? In terms of their own vaccination. It's really interesting. It's almost universal.
And I've heard a high school student very close to me tell me that girls would line up at school and they would get a shot and they might not even know what the shot was for, but they were getting the shot. So that was an idea of 100% trust. Why were they getting the vaccine? They weren't getting the vaccine because of knowledge or education. They were getting it because of trust. So, yeah.
Okay, I'm going to repeat the question for the people online. So
I'm a nurse at Sitges, and I have a niece who's an anti-vaxxer. I guess I want to know if you have therapy sessions for people.
For you.
No,
yeah, yes.
because everyone I work on a unit where we have immunocompromised children. We have kids with cancer, we have kids with transplants, and we know that those kids have died. And I get so angry that I know I can't even talk to my niece anymore. I'm actually avoiding her. And I'm too angry. I think I'm just going to choke her. You know, so that's not a good, it's not a good way to get across this, I'm sure. Do other people say that, or I don't know.
No. Oh, yeah, repeat the question. Go ahead.
okay, so I'm going to paraphrase. So basically it's, and let me know if I'm not articulating this well, but it's, and I think a lot of us feel it, it is is there any therapy sessions and, you know, just assistance for those of us who are very strong in our beliefs in immunization, dealing with anti-immunization folks, especially in social circles, but also in professional circles?
And my answer was just no. There's no help for you. For me, if I had to talk to somebody who was hesitant to vaccinate, I like to have a very simplistic approach. You know, being a man, I have a very simplistic approach to things. So I like to think of things as being in three groups. I think of the person who first may have a couple of questions. Is there mercury or thimerosal in the vaccine, and what's the effect of that? And then there's the people in the group that have been on the internet and have read and have made themselves knowledgeable and have lots of questions. And then there's the third group who might be in a group where they have religious beliefs that don't want them to vaccinate, that they have been part of an institution like alternative medicine in certain spots, that they won't vaccinate, and this is a very hard group. And when I come across a person in the third group who comes across me personally, I basically meet them and agree with them, everything that they have to say. Because again, from their perspective, they think I'm lying, I think they're lying. We're both lying to each other. And we're both on equal grounds. So I say to themselves, I don't have much to say to you because the evidence that I may have for you, you might not agree to that evidence. And I respectfully may not agree with what you have to say. And I find allowing myself to befriend and engage and again, to build a relationship with them. I'm a pediatrician, so I have these patients in my practice, and I build relationships with them. I just say, okay, and they're flustered by that. They don't know what to do. Now they have somebody agreeing with them. And then we move along as far as dealing with their kids are concerned. Of course, they're gonna get a fever, they might get an ear infection, they might get some asthma, they might get this and that, and now they realize that they may be starting to look for some answers from another viewpoint that may be able to help them. And inevitably, a couple of months goes by and we start talking about individual vaccines maybe, like Prevnar, for example, that doesn't seem to hit the literature as much as MMR and pertussis do. And then they start vaccinating with Prevnar, and now we have a trust because there was no reaction to it. So generally, that would be my approach, is actually to move that forward. Yeah.
Hi, thank you for your talk, Steve. I'm a family physician at North York General, and I have a comment and a question. One is, your talk blew my mind. Like, I think I live in a little world where I assume that all of my colleagues thought the same way as I did, and obviously not. So now I wonder if I need to go back to my group, do some data mining, and then invite you back for faculty rounds. But my question to you is that first study that you presented where we were looking at the HPV vaccination rates and the gender of the physician was the most significant factor. Did they look at other confounding factors?
Absolutely, they did, but that was the only one that stood out.
So they looked at things like how long the appointments were, how much time, like if these people were having a practice skewed towards women and children and how much time they spent within the visits, and the only thing that made the difference was the gender?
The only significant thing, one of the factors, I don't know if it was from that study, but there are differences in practices where there are many people who need to be vaccinated versus few people who need to be vaccinated.
Right.
So if you have a family, if your family practices of, you know, maybe elderly people and as opposed to all babies, that would skew it. So the more patients you have to vaccinate, I think that's like that in all of medicine.
Yes.
The more engagement you have with something, the better you are at it.
Yes,
so
that's
that was
what I'm
true.
wondering.
But it wasn't the single most important factor.
Okay.
The single most important factor for patients is healthcare providers' recommendations and the patient's concern about side effects. And for healthcare providers, it's their own concern about side effects, which is what we have to really delve into. And their own lack of trust in recommendations. So an interesting thing to do would be to see if somebody in your practice has a lower immunization rate, because I think you guys track that anyways.
We do.
Exactly.
So I know mine. Now I'm going to go check everybody else's. But anyway.
Exactly. So if you find somebody who has a lower immunization rate, maybe take a look at these things. The literature would support that there is provider differences would be number one about their own personal beliefs and attitudes towards immunization. And then practical things. The only really solid practical evidence I had was not giving multiple vaccines on a single visit. And that might be your red flag. So if you could see that somebody moves a vaccine from one visit to another, think about that. Why is that healthcare provider even doing that? Why are they changing the immunization schedule?
No,
there's obviously a thought in their mind, and they have concerns about that. And that, to me, would be a very easy thing to pick out. Yeah, nice.
I'll be emailing you.
Okay, well, we are at our time for a break. But before we go on a break, I'd like to thank all of this afternoon's speakers, and in particular, Carly and Stephen, for the last hour or so. You were both fabulous, and we all learned a lot. So thank you very much. Now we have a break until 3 when we will start the small group conversation about and speak some more about vaccine hesitancy. And I know people had some questions And both Stephen and Carly will be around for a bit of time to answer any questions you have. So thank you. Okay, for the next session, what we're going to be doing is having small group conversations about vaccine hesitancy. So you will see that there's 3 questions here that we want you to reflect upon. I'll tell you, this is, we're going to give you instructions about how to reflect. So, but these are what you're reflecting on. The first is, how can public health or scientists work more effectively with journalists, the media, and social media to address vaccine hesitancy? The second is, what is one way the Center for Vaccine Preventable Diseases address vaccine hesitancy in healthcare workers? And the third question is, please identify one way in which your practice or work or thinking will change as a result of what you've heard today about vaccine hesitancy. And how we're going to do this is, first of all, there's facilitators who will be floating around. They're either at your table now or they will be floating around, and I'm going to have the facilitators just stand up so that people will know who they are. Okay, so even though they're right there now, they're not going to stay there, they're going to float. And what we want you to do is, and you'll see that the questions are there again. So first we want you to think on your own for 2 to 3 minutes. And then we want you to share with your neighbor for 5 minutes. And then we want to share at your table for 10 minutes. And then we'll have people share with a room for 15 minutes. So this really should be think, pair, share, Michelle, wherever Michelle is. And so what I'll do is I will stop talking in a minute, and I will give you a notice when you should start the sharing at your table, and then come back for the sharing at, with the room. Okay, go. Okay, so it's time for our second share. So what I'm going to do is ask for volunteers now that the front table is quiet. We're going to ask for volunteers in the room to share with the bigger group either one question or more than one question. But one thing that we need to do is have you speak in the microphone. So Whitley's going to run around with the microphone to whoever to whoever wants to share first. So who would like to share? The front table, the talking table. Yeah, woohoo! Faster, faster. Okay.
Hi, everyone. I'm going to specifically talk about item number 2, which is how we can address vaccine hesitancy in healthcare workers. So we're building off of Stephen's talk here, which is targeting hesitant workers themselves so that they can then turn around and help their patients and lead the patients to the vaccine. So the idea that we're building off of is, I'm sorry, the tall gentleman from Australia, we're building off of your idea with the opioids that we were talking about, and it is finding the vaccine hesitant healthcare workers and going and engaging them in some way. So we've had an idea that we need ISIS involved, or we need really access to billing data to see who is actually vaccinating at the rate they're supposed to be versus the lower rate. Find those lower rate people and reach out to them. One way is that we can actually go and talk to them and engage them one-on-one, which is probably really expensive and going to take a long time. Another way to do it, maybe before we get to that step, is to let them know where they stand in terms of the pack of vaccination rates. So if you got an email or a printed letter, a scary printed letter telling you were 40th percentile for vaccination rates, or you're at a rate of 40% when you should be something much higher than that. Sometimes data can be a powerful indicator, especially in a competitive situation, looking at your peers. So, and then to take that one step further with the one-on-one conversations that Stephen talked about. That's all.
Great. That's great. Okay, do we have another volunteer? Oh. And I like that we do it to, I was going to do maximum.
Okay.
Run it for you. Okay, go ahead.
Thank you. That's a nice segue to what we were going to talk about for that question. What can the Center for Vaccine Preventable Diseases do? So one of the great things about this center is it's sort of being rooted at the university, where there's such a diversity of expertise. And so it's to engage the researchers to do exactly the sort of thing you were talking about, whether it's bringing in the social scientists, the psychologists, the behavioral scientists, to test those kind of things out, to help those of us in the field know actually how to implement it. So I think that's a great thing that the center could take on with its sort of foundation across the university.
That's a great idea. And do we, over to Chris?
Hello. So as I guess an historian amongst all of us, I'll just sort of emphasize, we've discussed this as well amongst us, but there's some real lessons to be learned beyond just the obvious lessons learned idea. There's more practical ways to approach things. I've been involved with what's called the quarantine tent, you might have heard about. It actively engages the public based on sort of time-traveling disease victims made up in a sort of theater, street-level thing. It works quite well. And I've yet to have a big argument with anybody in the midst of that. It's quite positive. But the point of that is kind of addressing those questions, is to personalize the history, in a sense, to give a real sense of what it was like at different times with different diseases. And it's also important to differentiate diseases. It's too easy to put them all in one group, but diphtheria is very different vaccine, very different disease than pertussis. They're opposites in many ways. One's extremely effective, extremely safe, but pertussis has been kind of a bitch of a disease all along, and it's been like that, still is. So, and also the key thing is to localize. There's a real story to tell literally right here, in terms of what most of these vaccines we're talking about wouldn't exist or wouldn't have been proven to exist without what happened literally at this university when Connaught Labs is a part of it. And there's also a continuity with that. Didn't stop at that point. It's still going on, not only with the concurrent incarnation of things, but other companies and other people. So don't be afraid to tell the local story. From a Toronto perspective, Canadian perspective, and do that in an honest way, emphasize the people involved, the science involved. I mean, you can't cherry pick science. It either works or it's not going to work. And so that's another way to engage the public, is to, so you're going to question your science behind your cell phone, anything else you can pick out of the air. It's all the same science. And also trying to get around the idea of the individualistic culture we have. That sort of ties in with the healthcare workers, I think, because they're very often adults, very individualistic, concerned about their particular situation, concerned about reactions. But they need to basically understand, in particular with flu, that it's not really about you. It's about the effect on the patients around you. And just to try to negate or get around that whole thing. And also for the third one, I just like the idea that came up in the last talk of more directly engaging the hesitant. Maybe not the very hardcore one, but your average hesitant parent, sort of head on. I just like that whole idea is just, because they're fellow human beings. Often there's personal situations that are informing them for their own child, whatever. But also, again, to emphasize what they do is much more than just what they do to themselves. It's there's impact beyond your own case. So anyway, that's kind of the gist of what we were talking about.
That's great. And I think even further, the one thing that we all agree on typically as parents, if we're talking about vaccinating children, is we're all in it for the best interest of the child. So finding a common ground, like Steven said, I think is an important thing to do. Next volunteer. Everybody looks down when you ask for a volunteer. Okay, we got one over here.
So at our table, we really only managed to get through 1 and 2, but we kind of combined our approach to 1 and 2. So what we talked about a lot was the online presence and the social media that goes into the vaccine hesitant or anti-vaccine websites, and how available they are, how engaging they are, and how easily accessible and user-friendly they are. And then when we compare them to the more factual websites, I'm sorry to say they're not as interesting, right? And so we had kind of talked about some of our own personal fears in engaging people online and what happens when we have done that. But this is where the Center for Vaccine Preventable Diseases might come in terms of creating a platform where, one, people who have had adverse vaccine outcomes, not adverse vaccine outcomes, but preventable vaccines, preventable diseases as a result of choosing not to vaccinate, if they wanted to speak up, they often feel suppressed online in these groups. And so providing them with a platform where they can share these stories so that others can learn from them, because the stories are very powerful, as we've talked about already today. So kind of creating that platform, but also creating a more interesting kind of online presence that is fact-based. Because most of, if they're very far on the end of saying, I'm convinced I'm not going to vaccinate, you have to take a different approach. But most of the patients that I see in my practice who don't want to vaccinate are in the middle. And they often come in saying, my friend told me, I saw it on Facebook, I read it somewhere, and then we've got to go and debunk that. So if we had a better online presence, then they might find our websites rather than those other websites, right? And the stories are really important.
That's great. Any other people willing to share? There's also been the other mom who has turned into a vaccine advocate that has become, that is now engaging in a lot of conversation, is the pertussis mom, who a couple years ago, I think she had 5 children, had just turned the corner to vaccinate, and they all got pertussis. And she was at a recent panel, the one that Teresa Tam led a couple of weeks ago, and was there as the mom talking and telling her story. And I think it's those sorts of stories, similar to the flu story that Carly spoke about earlier, those type of people that really help us sell our story, which is that vaccines save lives. Other tables? Other tables? Natasha's table of 1.
Given there's a gap, I did get contacted by a journalist a couple of months ago who said to me she wanted to know whether Public Health Ontario endorsed vaccines. And I said yes. And she said it was interesting. She'd rung up the Canadian Pediatric Society and a whole bunch of other organizations and looked on their websites, and she couldn't find anywhere. And she wasn't a health journalist, so she wasn't really, this wasn't her thing, but she was a local, I think, Ottawa Valley journalist. She was looking at, Public Health Agency of Canada, and I don't know if she looked at the OMA, but she was trying to go through all the kind of websites she could think of, and I couldn't find any of them that said, we support vaccination, as a kind of bold statement. And apparently CPS did have a statement like that on there at one point, but it sort of comes and goes, because the way you have to keep your websites fresh with this new story coming up and all that. Anyway, I just thought it was really interesting because it was something so simple that we could all do, that just to say, we support immunization. Any institution, any health-related institution, just to state that completely plain, loud and clear. Thanks.
Maybe part of our conflict of interest statement would be, I'm Shelley Deeks, and I support immunization. Then and we'll have a new movement for COI statements. Anybody else? Oh, we got one.
Hi. So one of the things that we talked a little bit about too was sort of in terms of training for healthcare providers is maybe sort of having some key messages when they're talking about vaccination. So instead of sort of maybe fumbling for, what thing would be the most effective towards this person, sort of having things that are kind of key sort of bullets that they could really talk to people about. And I guess we had a little bit of a discussion whether that really makes sense as far as, needing to tailor the message to the person's concerns and appealing on an emotional level. But I think one of the challenges that we talked about is that, you're in a busy, fast-paced clinical environment a lot of times, I think, and you don't necessarily have the resources and the time to stop and get into that emotional level, figure out what this person's, specific concerns are. But maybe, I guess, like, sort of to speak to Natasha's point a little bit about, just being sort of confident, like having that sort of confidence around at least those few key points. Because I sort of think back to even to Dr. Moss and thinking about providers being really confident in their recommendations. Like maybe the problem is, partially that we're not being sort of forward and confident enough on the pro-vaccine side, just in terms of without equivocation, like we support vaccines and these are the reasons why, and they're safe predominantly, and those types of things. So maybe it's sort of about a stronger message. I don't know. I know, I just put a caveat on it. So they're safe, period. Anyway.
Okay. I think we've got Irene up.
Thank you. When I was a resident here in public health and preventive medicine, we had a partnership with the Ryerson School of Journalism. And so we all had an opportunity to work with a journalism student in a print piece, in a radio piece, and a TV piece. So we would pick something and we would work on it, and then we would have one day where we all met as a group, and like for the print piece, I'd meet with the journalist student and they'd ask me a bunch of questions. I'd try and get across my message to them. And then it would happen with everybody. Then we'd all convene on a day where the print piece would go up and did my messages get across? And there was sort of an opportunity for feedback in both directions for those three different kinds of media. And I wonder if there's an opportunity for something like that to train healthcare providers. I know right now at Toronto Public Health we have ID fellows from both the adult and peds program on rotation with us, and they're all really happy to get some media training. They're like, yeah, we're worried that we're going to get called to do some piece when we're practicing, and this is really helpful to us. So I think more opportunity for actually working with media, training with media would be very useful.
Okay, we've had a lot on 2 and 3. What about the work more effectively with social media? Over here.
Okay.
Not over here.
Is that okay? Yeah. Oh, hi. This is my personal opinion. I just wonder, can an open forum be organized in certain communities to invite the public to come to listen to informed decision by some authority or something, rather than doctor or nurse to tell you, just one-to-one, which is a lot of time, How about that? It's just my opinion.
I think that's a very important opinion, and you would reach many more people in that way. One thing that was interesting when there was a recent discussion about engaging the roles that Google, Facebook, and Twitter are taking to counter vaccine hesitancy or vaccine refusal was the caution that we do not want to be seen as censors. And so, because that will just feed into the conspiracy theorists. And I don't think we often, we may not often think about that lens, and I think that's also an important perspective to ensure that we don't be seen as censoring with engagement with social media. At the back.
The thing that we talked about at our table, particularly on the media, was the concept of training, which I think Irene referred to, how important that is you know, particularly if you're in a small local environment and the small local media wants to talk to you. You know, you have no media training, and this is, you know, the same journalist that's covering the motor vehicle accident an hour later. But then also on the social media is trying to find this space for different types of organizations. You know, as a governmental organization, we're not particularly in that space because it's social, it's two-way, it's 24/7, it's rapid, fast. It's not how things work in my organization. But that's also, so where is the space for that? We were talking about that, so we didn't have an answer to that one.
Maybe the center can take that on.
Yeah.
I think there might be something over at, someone over at this table that wants to speak. Oh,
It's online, the online.
I'm just sharing some of the comments from online participants. There's some comments about in global health settings, particularly in Pakistan and Afghan refugee camps, sharing stories and using media advertisements as a way to promote vaccination, and clearly, but also one of the participants talked about the lady health workers going out to the community, because in that case it's not necessarily that the people are going always to the clinic, but rather that actually the lady health workers will be going to the families and parents to engage with them, and they played a major role in improving vaccination coverage.
okay. Well, if we don't have any more questions, or is there any more comments online? They were great, great comments. I'll bring this session to a close. I think it's been a really interesting session with lots of food for thought, and was very successful. So now I'm going to turn over to Dr. Michelle Murdy. Michelle is, I don't have your bio, Michelle, so I'm just gonna say it. So Michelle is a public health physician at Public Health Ontario with a lot of interests, including HIV, influenza. Actually, it's probably easier to list your non-interests. That's how many interests she has. And Michelle is going to announce the prize winner for the student presentation.
Well, thanks, Shelley. I think the reason I don't have a bio is because it's not about me. It's about congratulating the students, and I think that's the first thing I'd like to do, is to say thank you very much to the 6 wonderful student presentations that we had today. So I had the pleasure on the Vaccine Symposium's planning committee to help solicit the call for abstracts, and I can say we had a number of really wonderful abstracts that were submitted. And thank you very much to all the judges from Public Health Ontario who helped to review those abstracts, and a difficult job that they had from selecting from those to select the 6 presenters that we had today. And then we had the judges that were named earlier, thanking them again in terms of their very challenging task of listening to these 6 diverse and really fabulous talks that were given today to help choose both a winner for our session today and as well as a runner-up. And this is a challenging presentation. They have 3 minutes to convey what probably could spend at least an hour talking about their work, and you could see the passion and enthusiasm amongst all the students. So I did want to say thank you to all of them, and I would say our first runner-up is Corinne Moss. And I unfortunately think our winner today had to leave, and so is not here, but we will make sure that she is aware, and we'll get her prize as well. And our winner today is Melissa Perry. Thank you very much.
Okay, the students are just, all of you are amazing, and it just makes me feel very warm about the future. So It's my honor to bring today to a close. I'm just going to say a few words. Don't worry, it's not going to take very long. I think it's been an amazing day, and we've really spanned from the very basic molecular level of things all the way through to talking about what we think about vaccines and feel about vaccines, and what healthcare providers feel and think, and what journalists are picking up from that. So it's been an incredible span, and I think that reflects where we would like to go with the Centre for Vaccine Preventable Diseases. And we're only just starting. We've only just launched this idea, so we're actively fundraising and figuring out how we can make something really sustainable in the long term. I think the comments that I heard about social media and having somebody engaged in social media, we have started talking about that. I mean, these big rock star people, apart from President Trump, but most of the really big people who are on Twitter all the time have a full-time kind of Twitter person who's doing that for them. And people like myself who occasionally tweet, that's my kind of category of tweet. I do the occasional tweet, but it's something you have to go into seriously. So we're talking about it. But I do agree. And I wonder whether together we aren't all stronger. Like maybe it's the collective voice on social media that will make the volume loud enough to have an impact. I also really feel the world has changed. When I mentioned the UK, and the media there are really awful, like campaigning against vaccines. But even in Canada, when I first got here, I was really impressed by how good the journalists were and how they actually did want to get their facts accurate, and they didn't seem to have an agenda. But they weren't quite as welcoming when it came to vaccine stories, whereas in the recent media experience, I don't know if other people had the same thing. I mentioned a bad example earlier, but really, everywhere I go, journalists are now saying, I'm so glad you're speaking up for vaccines. Like the tenor has changed, and I think that probably reflects something else that's going on in society, that people are talking about vaccines differently now. So I see that positively. There are an enormous number of people to thank for today, and I don't want to go through naming everyone, because I think lots of people at Public Health Ontario have helped with today. There was one person who I really want to thank for today, though, in particular, and that's Dr. Shaza Fadel. And I hope everybody else who's been involved with organising this day recognize that she has been the person who really made it all happen. And she's at the back there. Could you stand up, Shaza, please? What's going to make this centre fly is the faculty and the students. So I hope that you will all support, sign up, contribute, help us figure out how we can make this fly and become a sustainable centre for Ontario, which has had this gap. We are the biggest province in Canada, and we didn't have anything. So now we have something to start with. In fact, with Shaza's efficiency, the evaluation for today is in your inboxes. So it should be. If you haven't got it, then let us know, but we've sent it out already. And when you do your evaluation, you will see there's also a link to the U of T, to the pages that we've got going for the centre, and you can sign up for the centre there, which means, you get early bird reminders about the next Vaccine Sciences Symposium, which I think we may well have to have in a bigger venue. So, thank you all for coming. Big thanks to all our supporters here in the room and outside the room. I hope you enjoyed the day, and it's been wonderful to see you all, and safe journey home, everyone. Thank you.